Faith. Okay, sorry, Faith. Okay. Sorry. Paki-join na lang, Faith. I don't know, weird ha, Google Meet ang sa'yo panin, it's not me. Okay, all right. So, ayan, so we'll continue. I believe the children is our future. Charet. Okay. Okay, so I believe, sige, diri ta mag-start because I think wala ni sa presentation gahapon. So this is a comparison between Basilary dysentery and Amoebic dysentery. Okay, now Basilary dysentery is of course, there is a need no for us to differentiate the two diseases because both of them are dysentery, no, na diseases. Okay, so both of them are dysentery, so we need to differentiate the two because again, they could be of different causes, no, or different causative agent ang nag-cause sa imong dysentery. So it could be bacillary or amoebic. So of course, amoebic dysentery is caused by Entamoeba histolytica, and most of the patients that are infected with Entamoeba histolytica, 'di ba, majority ang symptom kay amoebic dysentery. Now for bacillary dysentery, this is caused by of course bacteria, your gram-neg bacilli, no, enterics, usually si Yersinia, Campylobacter, Shigella, Salmonella, and enteroinvasive E. coli or EIEC, EIEC, no? Ay AYEC, di AYEC. Okay, so kana sila. No, so we have to differentiate para at least makabalota kung kinsa by causative agent. So for bacillary, it could be epidemic, no? So pwedeng daghan ang ma-infect. For amoebic, seldom lang siyang epidemic. So usually gamay lang. For symptoms onset, no, mas paspas ang bacillary, acute. For amoebic, it's gradual, so medyo slow. All right. Now there could be a prodromal fever and malaise sa imong bacillary dysentery, so meaning na ay ginagmay na ma-feel na fever and body aches imuhang patient. For amoebic dysentery, wala yung mga prodromal features. Vomiting is common in bacillary, sa amoebic dili. Patient is prostrate, so meaning dili siya katindog or maglisod siya og go about sa ihang daily life. Whereas for amoebic dysentery, your patient is ambulant. So pwede pa siyang stand up, no? Pwede pa siyang maglakaw-lakaw, maglaag, ganun. And for the diarrhea, watery, bloody mo ang bacillary, and for amoebic, bloody. Okay, now for the odor of stool, odorless mo ang bacillary, amoebic is fishy odor. For microscopy, again, very important, of course, sa bacillary, you can see a lot of gram-neg bacilli because again, this is caused by bacilli, no? And then you have also pus cells, no, RBC, red cells, yes, pero no charcot-laden crystals. Okay, now for amoebic dysentery, stool microscopy, few bacilli, no, mga gram-neg rods. Trophozoites, of course, because this is considered or this is caused by an amoeba, no, amoebic. So you can see trophozoites, so very important. Trophozoites ingested with our trophozoites with ingested RBCs and the presence of charcot-laden crystals. So kinsa din yung charcot-laden crystals? Unsa man ni sila? Your charcot-laden crystals are breakdown products of what type of cell? Kakinsa man ning cell? Ang charcot-laden crystals. Breakdown products ni? Yes. Okay. AUBF, kumusta tayo dyan? Okay. Or HEMA. Okay. Charcot-laden crystals. Breakdown products ni kinsa man na cell? Your kinsa man? Kakinsa man yung cell? Ang breakdown.
Basilophill, sir.
Okay, all right, that is correct, no? So it's the breakdown products of eosinophils. Kinsa to maglahi ito siya natingog. Parang bago yun.
Section H, sir.
Section H. Okay, very good. So, that is correct, no? Don't forget, eosinophils, hang yung breakdown products kay charcot-laden crystals. So, makitaan gyud ang charcot-laden crystals sa amoebic dysentery because, you know, parasite, no? Parasitic disease ang imong amoebic dysentery. So, generally, gyud, di ba, we say that eosinophils are increased, especially during parasitic diseases. Okay, but not all parasitic diseases have increased eosinophils. No, general lang. Most of the parasitic diseases, they will increase your eosinophils. All right, so amoebic dysentery, so na charcot-laden crystals. Nakawag na maisap ha, basta charcot-laden crystals, that's from eosinophils. Hala, yes, okay, all right. Now, abdominal cramps, common and severe sa bacillary, pero not common siya or mild lang sa mukhang amoebic dysentery. Now, tenesmus is common among bacillary dysentery. Tenesmus is the constant urge, no, the constant feeling to defecate or to poop, no. So it's common in bacillary, not common in your amoebic dysentery. Now, for recovery, medyo spontaneous ang bacillary, and the chances or the of remission or relapse is not common, okay? Wala siya relapse. For amoebic dysentery, pwede siyang magka-relapse. So, pwedeng ma-infect na po doon ang patient. Alright? So, in general, as you can see, di ba, ang symptoms sa bacillary dysentery ang medyo intense compared to amoebic. But, as you can see, uh, mas wala siya chances of relapse, no? Mas wala chances na mabalik siya usog. Okay? All right. For amoebic, pwede pa siyang mabalik, no? And mo persist pag yun ang infection for years. Ayan. So pwede siyang mudugay. Sana all. Charot. Okay. Now, of course, very important difference is microscopy. The presence of trophozoites, no? In amoebic dysentery, and of course, numerous gram-negative bacilli in your bacillary dysentery. And of course, charcot-Leyden crystals in your amoebic dysentery. Okay, so ayan, bacillary dysentery, di ba? So we mentioned that it can also be caused by enteroinvasive E. coli. So ayan, so enteroinvasive E. coli is a biotype of E. coli, di ba? Daghan man to sila. You have enteroaggregative, enterotoxigenic, you have hemorrhagic. Ayan. So we'll focus on EHEC. Okay, siya may pinaka common or popular. Charot. So for EHEC, no? EHEC. Unsa man na serotype ni si EHEC? Your enterohemorrhagic E. coli. Unsa ba ni na serotype? Okay. Ayan. Kakinsa man ni? Kinsa man ni na ano? Unsa man ni na serotype ang na E. coli ning EHEC? E. coli, unsa man? E. coli blank. Oh, ayan.
O157H7.
Okay, very good. No, E. coli O157H7. So that's your EHEC. Now, if you want to isolate EHEC or E. coli O157H7, unsa man to atong culture media of choice for this organism?
Sorbitol, sir.
Sorbitol, unsa man? Sorbitol ra? Sorbitol.
Makonki agar.
Okay, all right. So our media of choice for EHEC or E. coli O157H7 is your SMAC, no? Sorbitol maconchi. Now, in sorbitol maconchi, unsa man atong ginalok na mga colonies, unsa man na color or appearance sa colonies para makaingon ta na E. coli O157H7 na siya.
Metallic green.
Colorless.
Metallic green? Okay.
Colorless.
All right. Colorless. All right. Sige, unsa pa man. Napailang answers? How about pink? Di ba siya pink sa SMAC? Or di ba siya yellow? Alright. So, ano itong final answer? Colorless, green metallic sheen, or pink or yellow? Unsa man? So, ano itong final answer? Colonies of E. coli O157H7 on SMAC. Unsa man? Sa itong answer? Final answer na to. Aray. Yeah.
Thanks, sir.
Pink? Okay. Yeah, sige.
Colorless, sir.
Colorless. All right, final answer na gyud ang colorless.
Yes, sir.
Okay. All right. So the correct answer is sa SMAC, your E. coli O157H7 have colorless colonies. Please take note of that. Do not forget because your E. coli O157H7 do not ferment or does not ferment sorbitol, diba? The rest of your E. coli subspecies serotypes, they ferment sorbitol. That's why colorless siya sa SMAC. Very important. Okay, all right. So green metallic sheen, and the answer green metallic sheen, that is the characteristic appearance of E. coli colonies on EMB. Okay, so your eosin methylene blue agar, green metallic sheen. Okay, green metallic sheen na colonies on EMB agar presobuzzer sa E. coli na siya. But ang atong question man is E. coli O157H7. So, di ba you've answered? Media of choice is SMAC, sorbitol maconchi. That is correct. Pero ang question now is what is the expected colonies of your E. coli O157H7 on SMAC? The answer is colorless. Okay, please take note. Yes, mga ano na, mga presobuzzer na mga questions. Okay? All right, ayan. Sige, so again, that's the comparison between your bacillary and amoebic dysentery. Okay, all right. So we'll continue. All right, so I believe kani dirita ni end sa last lecture, no? Kani trophozoite sa ihis. Okay? All right, ayan. So we'll now proceed with the next Entamoeba species, and that is your Entamoeba dispar. Now, Entamoeba dispar, very important, is that this type of Entamoeba species is morphologically identical with Entamoeba histolytica. But this type of species is not invasive and it's not capable of causing disease. So basically, morphologically identical sila with E hist. So in a way, twin ni siya, no? Twin sila ni Entamoeba histolytica, but this one is the more kind, no, or the good twin. Whereas si E histolytica siyang evil twin, ganun. Okay? So it's morphologically identical with your E hist, all right, but it's also the more common species, but it's incapable or it's non-pathogenic. Okay. And because it's morphologically identical, no, you cannot really distinguish the two by morphology alone. And if you want to speciate, you must perform mga DNA probes, no, mga molecular techniques and electrophoretic techniques to isolate mga enzymes, to isolate enzymes that are specific in each na species. Okay, so that's for speciation. And usually, speciation happens in mga high-end laboratories, no, research reference laboratories. But in routine hospital setting, let's say you saw a trophozoite, no, while examining a stool sample, and the trophozoite contains ingested RBCs, okay? It contains ingested RBCs, then you have to report it as Entamoeba histolytica alone, okay? Because again, characteristic yun sa trophozoite sa E. histolytica ang present sa ingested RBCs. Your Entamoeba dispar trophozoite does not ingest RBCs because again, it's not pathogenic, no? But in routine hospital, as per experience then, that's quite difficult to see under wet preparation, especially during direct fecal smear. So that's why if we see a trophozoite that could cause or that is causing the patient's disease in a sample, we report that as Entamoeba histolytica slash Entamoeba dispar seen. Okay, so you put Entamoeba dispar, okay, because again we're not sure if it's really E hist or the E dispar. So to be safe and to be much more accurate, no, we report Entamoeba histolytica slash Entamoeba dispar. Okay, but in the case na you performed siguro permanent stained smearing, no, or you saw ingested RBCs in the trophozoite, then presbyopia inyong i-report lang siya as Entamoeba histolytica. Okay, all right, but morphologically identical, ang cysts the same, trophozoites pod same, all right. But again, presence of RBCs, that is diagnostic or that is really specific for Entamoeba histolytica. Because your Entamoeba dispar, again, trophozoites do not ingest RBCs. Okay, all right. That's for Entamoeba dispar. Next, you have Entamoeba Moskovski. Your Entamoeba Moskovski is another parang it's another species that looks the same with your Entamoeba histolytica. But this Entamoeba was first detected in sewage, so sa mga wastewater, no? But can also be found in mga rivers, sa sediments sa rivers, and also sa brackish coastal pools like kanang kung asa mag-meet ang water sa river and sa sea, parang ganun. So brackish area, okay? Again, it's morphologically indistinguishable from the two, Entamoeba histolytica and Entamoeba dispar, and also again they differ biochemically and genetically. So if you want to perform that, again, mga high-end laboratories ang na capability to perform such tests, okay? But routine hospital usually medyo wala, okay? So we don't have the capability to speciate. Okay, all right, ayan. Now, it's physiologically unique because it is osmotolerant, meaning it can tolerate high concentrations, and it can also live at room temperature and even high temperatures such as up to 41 degrees Celsius. Okay, all right, ayan. So that's the difference between E. Moskovski and the rest, E. histolytica and E. dispar. But again, it's generally considered to be non-pathogenic. So silang tulo, no, the three, Entamoeba histolytica, Entamoeba dispar, and Entamoeba Moskovski are really morphologically identical with each other. Okay, so for reporting, usually if mo report in hospital setting, kutob ra gid siya Entamoeba histolytica slash Entamoeba dispar. We don't include na Entamoeba Moskovski because again, quite rare naman siya to recover, and Entamoeba Moskovski is understood to be a part of the E hist slash E dispar na group. Okay, pag-reporting. All right, so it's not necessary that we include E. Moskovski during reporting. So again, routine hospital, no, as per experience, most of the hospitals, again, they report it as E hist slash E dispar trophozoite scene or cyst scene. Okay, all right, again. But again, if you see ingested RBCs in the trophozoite, then you report it as E. histolytica C. Okay, but again, depende gyapon sa SOP sa laboratory. Okay, so still follow SOPs, still follow the standard operating procedures in your institution. Okay, all right. So that's Entamoeba Moskovski. All right, okay. So detected in sewage. Next, you have Entamoeba Heartmanite. Ayan. So si Entamoeba Heartmanite is we consider as the small race, no? Small race of Entamoeba histolytica. So siya ang smally na version, di ba? Siya ang baby version ni Entamoeba histolytica. Okay? So wherever E. histolytica is found, you can also see Entamoeba Heartmanite. But again, it is considered non-pathogenic. Okay. Now for the trophozoites, again, as you can see, sa size pa rin, nagkakatalo or sa size tayo mag-base, no? It's smaller than your E. histolytica. So the trophozoites of your E. Heartmanite, as you can see, is 8 to 10 micrometers lang. For your E. histolytica, ang average is about 15 to 20. Okay, so nuclear cytoplasmic characteristics similar to your E. histolytica, but the peripheral chromatin has a beaded appearance. So I think wala na ako nabutang dala dears. So please take note lang. Again, beaded appearance sa peripheral chromatin. And again, of course, trophozoites do not ingest RBCs. Okay, so kanadjong presence of RBCs again within the trophozoite is diagnostic. Okay, is specific for E. histolytica. Okay, all right, ayan. Again, this is the small race of E. hist. So here's a picture of the trophozoite. So as you can see, diba, by looking at it, same appearance with the trophozoite of your E. histolytica based on nuclear and cytoplasmic characteristics, diba? Sa nucleus, you have a central karyosome, spherical nucleus, and even chromatin. Okay, and as you can see, the cytoplasm is also quite even. Okay, or fine rapod siya, finely granular. But again, the difference lang is the size. So as you can see, 10 micrometers, no? So it's smaller. So that is Entamoeba Heartmanite. Okay, so sa size tayo magbe-base, okay, or size matters aning Entamoeba Heartmanite. Okay, so does size matter in reality? I think dili naman. Charot. Okay, so ang important pa rin is performance. Check out, check out. Okay, all right. So, but for Entamoeba Heartmanite and Entamoeba histolytica, size matters. Okay, all right. Ayan. So, that's for the trophozoites of Entamoeba Heartmanite. Now, for the cysts of Entamoeba Heartmanite, ayan. So, the range is about 6 to 8 micrometers. So, muna siya inigawa sa inyong pre-test, di ba? So, gi-describe ang cyst, and then ang size niya is about 6 to 8 micrometers. Ayan. Now, it consists only of 1 or 2 nuclei. Chromatoidal bars, again, nuclear characteristics, cytoplasmic characteristics similar to E. histolytica, but the chromatoidal bars of your Entamoeba Heartmanite are more numerous and they are smaller. So, mas gamay sila and mas daghan. And aside from that, they have the ability to retain the chromatoidal bars when they grow old, diba? Because majority of the chromatoidal bars of your cysts, they vanish once your cysts become mature or once mugulang na imuhang cysts. Okay? All right. Now again, as mentioned, size matters for Entamoeba Heartmanite and Entamoeba histolytica. So dira ta mo base sa size. So here is an example of the cyst of your Entamoeba Heartmanite. So as you can see, same appearance pa rin with your E-hist, no? Nucleus, central karyosome. Okay. Now, di ba, based sa inyong pre-test, gi-mention dito na a cyst was recovered that measures 6 to 8 micrometer. Okay. Then, ang iyahang description sa characteristics sa nucleus and sa cytoplasm, similar sa Entamoeba histolytica. Di ba? So, if ikaw, nalimot ka sa size, giyod, sa duha, ako ato, personally, I would answer E. histolytica because that's the closest answer that I can think of. But now that we know kung sa mga size ranges nila, so ato na lang technique ana dears is if mobalik na po ito na question, basta gani ang measurement sa cyst is less than 10, okay? Less than 10 micrometers, E Heartmanite to siya, okay? So Entamoeba Heartmanite to i-answer. But if ang cyst kay greater than 10 micrometers ang gibutang, then ang atong i-answer ato kay Entamoeba histolytica. Okay? That is again if na ay given a size sa cyst. So usually if given na gyud ang size sa cyst, it would tell you na okay, ganahan ning board of examiner or ganahan nag-question ani na makabalo jud ko or ganahan siya na specific yun ang i-answer. Okay? But if walay size na given, no? So most probably ang imong i-answer ato kay Entamoeba histolytica. Because again, the general characteristics na gihatag ato for Entamoeba histolytica gyud to. Okay. So gihatag na to siya size para at least maka-think mo ba, pampa-confuse to siya, o di ba? So again, look at the size of the cyst ha. Kung cyst ang given is less than 10 gani, if less than 10 micrometers ang size sa cyst, then that's Entamoeba Heartmanite. But if it's greater than 10, that's for Entamoeba histolytica. Okay? So size matters, no? Size matters between your Entamoeba Heartmanite and Entamoeba histolytica identification. Okay? All right. Ayan. Sige. Next, for to differentiate the two, again, so this is your E-His na cyst and this is E-Heartmanite na cyst. So based on picture alone pa lang karon, so medyo makita nyo mo na dako, no? ang E-His na cyst. But it could be that na zoom ni siya pag picture, okay? Nya kaniwala. So it's important we measure it through micrometry, okay? So we measure it through micrometry. So as you can see, medyo same ra gid sila appearance, but for this particular picture lang, na timing nga na naapay chromatoidal bars ang E-histolytica. Okay? But again, naay times good na wala, no? Dili maklaro. So it's important good that we measure or we use micrometry. But in routine hospital setting, usually micrometry is not performed. Okay? So speciation between the two usually kay dili good mahimo. Okay? But if you want to speciate the two, okay, if you want to speciate the two, then you have to send your samples to your reference laboratories or mga high-end laboratories that could perform that. Okay? All right. So in routine hospital setting, it's already enough, okay, that we identify it as EHIS, cyst, no, slash EDIS parva. Okay? Because again, in a way, same ra po silang mode of treatment, diba? So, ayan, ganun. Okay? And E-Heartmanite is, of course, non-pathogenic din naman. So what we are really after is the pathogenic one, which is Entamoeba histolytica. Okay? All right. So again, for speciation, no, we use micrometry because size is what we base on its identification. Okay. But in routine hospital setting, again, micrometry, which is the which is how we measure, no, your cysts and trophozoites and protozoa. Micrometry is not routinely performed in your hospitals. Okay? Ayan. So that's for E-Hist versus E-Heartmanite. E-Heartmanite, again, is a small race of Entamoeba histolytica. So kato sila, previous species, Entamoeba dispar, your Entamoeba, ano to sa? Moskovski ay, no? And then Entamoeba Heartmanite. Sila ni siyang mga pare-parehas og itsura or they look the same with Entamoeba histolytica. Okay. And the next one is your Entamoeba poleki. Now, Entamoeba poleki is considered to be a parasite of your pigs and monkeys. And in some areas of the world, such as in Papua New Guinea, this is considered to be the most common intestinal amoeba of humans in Papua New Guinea. Okay? But in the rest of the world, the rest of the parts of the world, this is rare. Okay? Dili kayo siya makita. Alright? But in Papua New Guinea, very common siya. The most common intestinal amoeba dito. Okay, all right. Now, again, same with your other amoeba na species, Entamoeba species, ingestion pa rin ang pinaka-common na mode of transmission. Ingestion of, again, food, water that is fecally contaminated that contains your cysts. Okay. It could be human to human, fecal oral, carrier ba, asymptomatic, or from pigs to humans. Okay, so that's the modes of transmission. Now, ang trophozoite niya is similar with E. coli, Entamoeba coli. Okay, that is sluggish, meaning hinay siya mulihok. And at the center of the nucleus is a small karyosome. So, na small karyosome. Okay, all right. And comparing it with your Entamoeba histolytica cyst, the chromatoidal bars are angular or pointed. Okay, because again, ang chromatoidal bars imong E-histolytica cyst is again coffin shaped, no, or cigar shaped. So please take note. Coffin shaped or cigar shaped ang chromatoidal bars imong E-histolytica, 'di ba? Coffin, murag lungon, or cigar, C-I-G-A-R, cigar shaped. Okay. And aside from that, the cyst of Entamoeba poleki is consistently uninucleated, meaning usa ra gyud ka nucleus. All right? Whereas ang imong E-histolytica, pwede siyang mabutog 4 ka nuclei, di ba? So ayan. That's the differences of the two. Okay. Again, most common intestinal amoeba in Papua New Guinea, but in the rest of the world, medyo rare na siya. Okay, all right. Now, here is an example of your cysts, Entamoeba poleki. So, as you can see again, angular or pointed, iyahang chromatoidal bodies. Okay. And you also have here the trophozoite, which contains a small karyosome. Di ba? Medyo gamay. Dili na siya makita. Okay? So that's for Entamoeba poleki. So if nagkalisod-lisod na ka, di ba? Nagka-poleki na ka. Yes. Okay. So nagka-Entamoeba poleki na ang tanan. Yes. Okay. Ayan. So that's for Entamoeba poleki. Okay. All right. Ayan. So before we proceed to the non-pathogenic intestinal amoebae and also Entamoeba gingivalis, okay? Do you have any questions, dears, before we continue with the non-pathogenic? Sorry, excuse me. Okay. All right. Again, kinsa tong amoeba na to na na-detect sa sewage, okay? And it's osmotolerant, makaresist siya mga high concentrations, but it's morphologically identified.
Moskovski.
Okay. All right. Okay, very good. Okay, very good. Entamoeba Moskovski. Okay. All right. Next, you have again the small race of Entamoeba Hesulitica.
Heartmanite.
Okay. Entamoeba Heartmanite. Okay.
Hartmaning.
Okay.
Hartmanai.
Okay, very good.
Hartmanite, sir. Hartmanite.
Okay, very good. Entamoeba Heartmanite. And again, most common intestinal amoeba in Papua New Guinea, you have
Nagkapuliki.
Okay.
okay. Entamoeba Poleki. Taro nga ha, nagka-Poleki, yes. Entamoeba Poleki. Kinsa nitong most common agent na to that can cause congenital infection? Okay, remember pa ba?
CMV.
Cytomegalovirus.
Okay, ayan. Dapat na nakapaspas no CMV. Okay, very good. Okay, so again, if you don't have questions, we'll proceed now with the non-pathogenic intestinal amoebae. Okay, so we'll start first with its life cycle. So as you can see, no, ang life cycle gud sa amoeba, mapaihis ba na siya or the non-pathogenic, it's really, really straightforward and very simple. Dili siya complicated, iyang life, no? So, igora yung makaon siya, makaon ang cyst sa host, and then the cyst becomes trophozoites, multiplies within the intestines, and then release trophozoites and cysts, which can be detected in stool samples and can be a source of infection put for others. And again, can be a source of infection for the others, and then the life cycle goes on, no, by BTS charot. Okay, so ayan, so ina na siya ka simple, no, simple ra gyud siya kaayo. Unlike lang, ang difference lang between the non-pathogenic and Entamoeba histolytica na life cycle is that the Entamoeba histolytica is capable of invading other organs. Okay, it can go to other organs, sa brain, no, sa lungs, sa liver, 'di ba? Kay laagan ni siya na pagka-amoeba. Well, the rest of your non-pathogenic intestinal amoeba, katuni silang mga strict og parents, no? So, mag-live lang sila sa ilahang house, which is your large intestine, no? Or sa intestinal tract, no? So, dili sila mo spread to other organs, okay? So, dito ra gid sila forever. And of course, it does not cause any disease, okay? It's non-pathogenic, all right? Okay, ayan. So, inara siya ka-simple, no? So, may pag na-amoeba na lang ko, kaya nila ka simple yung life, no? Mahimong cyst, kanon pa jud ka sa host, no? Sana all, charot. So, inara siya ka-simple ang life. Sana all. May tag na amoeba na lang jud ko. Okay, ayan, charot. So, that's the general life cycle of your intestinal amoeba. Again, similar with Entamoeba histolytica, the difference lang is Entamoeba histolytica, aside from, again, colonizing the intestines, it can spread or it can invade other organs. Okay, all right. Ayan. So we'll start first with the first non-pathogenic intestinal amoeba, your Entamoeba coli. Now, Entamoeba coli, di ba, we have compared, no, Entamoeba histolytica sa first part, no, gahapon na lecture, if you can remember. So we'll continue again, no, review lang. So for the cyst, it's quite large, 15 to 25 micrometers ang size, spherical or oval, could be triangular or other shapes. Ang iyahang shape, okay? Chromatoidal bars, splinter-like, or it has a broomstick appearance. Ayan, please take note of that, dear. Murag wala na ako nabutang. Broomstick appearance, no? So, murag siyang silhing na ay mga murag pointed ends ang iyahang chromatoidal bar. So, splinter-like, broomstick appearance. Mature cyst, 8 nuclei, but some nuclei can have 16 or more, no? So, ina nakadaghang nuclei. Peripheral chromatin is coarsely granular and unevenly arranged. Ayan, so daghang granules, coarsely granular, unevenly arranged imuhang peripheral chromatin. So imuhang cytoplasm kay medyo dirty lang tawon, medyo hugaw. Karyosome is large pero it's eccentric, meaning off-center, wala siya natunga. And glycogen diffuse but may be absent in mature cysts. So makita na sa mga young cysts. So here's an example of your E. coli na cyst, no? As you can see, daghan siyang nuclei. And then here is using iodine stain, di ba? As you can see, daghan kaayahang nuclei. Usually, if greater than 4 na siya, then that's Entamoeba coli. So 5 kabuok ni, no? And this is the stained version. So as you can see, the cytoplasm is granular, di ba? Medyo hugaw, daghang granules, alright? And the karyosome, kanina sa tunga, is off-center, eccentric. Okay, so that's for the cyst of your Entamoeba coli. And if there is again chromatoidal bars, splinter-like, no, murag broomstick, murag silhig na tukog. Okay, ayan, broomstick appearance. Okay, all right. Now that's for Entamoeba coli. Okay. Now we have here again your Entamoeba coli na trophozoite. So for the trophozoite, quite large pa rin. it moves in several directions, okay, and sluggish, so medyo hinay iyang movement. Okay, now several directions siya because ang iyang pseudopods, daghan siyang pseudopods na ipagawas sa iyang trophozoite. So because daghang pseudopods na ipagawas, pwedeng daghang directions iyang pwedeng adtuan. Kay daghan man siyang pseudopods na gipagawas, so daghang false feet na pwedeng maglihok-lihok. So it can bring it to different directions. Ayan. And ang pag-thrust or pagpagawas niya og ay, bastos. Ang pagpagawas niya og pseudopods kay slowly. Okay? So dili siya pas-pas magpagawas og pseudopods. Unlike sa imuhang Entamoeba histolytica na explosive, no? Like pas-pas kayo mong pagawas og pseudopods imuhang Entamoeba histolytica. If maklaro ninyo tong video, siguro ako ipakita na lang karon before we end kay basta dili klaro. So nato yung video dito, di ba, of an Entamoeba histolytica trough na akong nakitaan while I was in Galliares, no? And then, if makakita mo sa iyang pseudopod, it's explosive. Paspas iyang pagpagawas og pseudopod. Okay? Alright, ayan, sige. So that's for Entamoeba Coli trophozoite. Endoplasm, similar with your cyst, dirty, no? May contain bacteria, yeasts, and cell detritus. Uneven distribution pa rin of chromatin. Again, coarsely granular. And of course, cardiosome is still eccentric, meaning off-center. And if there are RBCs in the intestinal tract, pwede ni siyang kanon po sa imong E. coli. But this is rare. It's not common. Okay. So, majority pag yung Japon sa yung cytoplasm kay mga bacteria, mga cell debris, and mga yeasts. Okay. And if the patient also has co-infections or other diseases like Giardia lamblia, pwedeng makaon po sa Entamoeba coli na trough ang cyst sa Giardia lamblia. Or also Sphaerita spp. Sphaerita spp is a fungus that is a parasite then. So it's a fungus that lives within the Entamoeba coli na trophozoite. So it's a parasite of a parasite. Parang ganun. Okay, but again, rarely seen siya, rarely recovered. Okay, most of the time, again, ang makita na nato kay mga bacteria, yeasts, or cell detritus ay ang cytoplasm. Okay, all right. Now, this is the illustration of the trophozoite. Ayan, and this is the picture. As you can see again, this is the the nucleus. Caryosome is off-centric, di ba? Wala siya natunga. Okay, off-centric. Again, the cytoplasm is granular, very dirty lang tawon. And you have here the vacuoles, kani mga circles dire. Okay? Alright, ayan. And uneven ang peripheral chromatin, meaning dili smooth lang tawon ang sulod sa nucleus. Okay? Alright, so that's for Entamoeba coli na trophozoite. So kabantay mo no, ang description niya, is opposite sa description sa Entamoeba histolytica. Di ba? Entamoeba histolytica, ang description nato, peripheral chromatin is evenly distributed, your caryosome is centrally located, no? Finely granular cytoplasm. So ang Entamoeba coli, opposite. Coarsely granular cytoplasm, no? You have eccentrically located caryosome and you have coarsely granular uneven peripheral chromatin. So opposite siya sa description sa Entamoeba histolytica. So if given ka na question sa imuhang exam gani or wherever nya, opposite siya, mga coarsely granular, hugaw lang tawon ng cytoplasm, daghang mga eccentric caryosome, then that could be Entamoeba coli. Okay? Alright, ayan. Now, this is a picture of Entamoeba coli with your sphaerita, as I've mentioned. Again, rarely seen, but this is the appearance. So these are kaning mga circles here or mga dots. These are the spores, no? The spores of Sphaerita spp. Because again, Sphaerita spp is a fungus. So pwede siyang reside within the parasite. So it's a parasite of a parasite. Excuse me. Okay. All right. So that's for Entamoeba coli. Okay, all right. Ayan. Next is your Endolimax, Nina. So if you can recall sa lecture gahapon, if naglantaw man mo, okay, Endolimax and Amoeba, diba? These two genera of amoeba, usually, they do not contain peripheral chromatin nor chromatoidal bodies. So if ako pa lang, no, makakita na kog amoeba na walay peripheral chromatin or chromatoidal bodies, then that could already give me an idea na, okay, this could be Endolimax or this could be Amoeba. All right, so we'll start first with Endolimax, Nina. So your Endolimax, Nina, it shows more nuclear variation than any other amoeba. So pwedeng maglahi-lahi iyahang itsura in terms of the nucleus na characteristic. So it could mimic, no, pwede niyang suhiron si Entamoeba fragilis or Entamoeba Heartmanite. Okay. Now, for the cyst, again, the size, kabantay mo, 6 to 8 pod, same siya with Entamoeba Heartmanite, but the shape is different. Majority of the shapes ay muhang Endolimax nina kay ovoid or ellipsoid. Okay, iyahang shape. Okay. Now, nuclei could reach up to 4, but ang pinaka-common na form is 4 ka-nuclei. So kani palang daan, different na sila with Entamoeba Heartmanite because your Entamoeba Heartmanite 1 to 2 nuclei lang. Okay, ayan. So that's for another difference, no? And aside from that, caryosome, blot-like. So when you say blot-like, it sounds wrong, charot. Ayan, so blot-like caryosome, similar siya appearance sa ball pen na nagblot sa paper inana. So blot-like caryosome and centrally located siya. And of course, Endolimax, no, genus, pag Endolimax gani walay chromatoidal bars and peripheral chromatin. Okay, so here's an example of your cyst illustration, diba? It's oval, ellipsoid, no peripheral chromatin, blot-like, okay, blot-like caryosome, rag hidden Mickey, charot. And then the cytoplasm is also quite granular. So here is an example of your cyst, no? So again, 2 nuclei, all right, 2 nuclei. And then as you can see, di ba, medyo dili klaro na ang nucleus because the caryosome is large, blot-like, okay? And of course, granular ang cytoplasm. Overall shape, again, ellipsoid or medyo oval. All right? Okay, ayan. So that's for the cyst of Endolimax niina. And for the trophozoite, again, 8 to 10 micrometers usually ang size. Sluggish, non-progressive, so hinay Japon siya move with blunt hyaline pseudopods. So blunt meaning dili kayo sharp ang edges sa imuhang pseudopods. Caryosome is still the same, blot-like, no, irregular, large, and absence of peripheral chromatin. And of course, cytoplasm, granular, vacuolated that may contain bacteria na yung ipangkaon. Yes, okay. So here's an example of the trophozoite. Again, granular cytoplasm, vacuoles, large, blot-like caryosome, okay, blot-like, ni-spread siya, okay, and again, no peripheral chromatin. So here's an example. As you can see, di ba, cytoplasm is granular, and the center is the large caryosome, blot-like, no? And kabantay mo sa kilid sa caryosome kay limpyo ra siya lang tawon because wala siya peripheral chromatin. Okay? So wala mo kabantay na murag na stain ang kilid, no? Sa imuhang or walay dark staining sa kilid sa imong caryosome kay again, no peripheral chromatin ang imong endolimax. Okay? But large yung caryosome, di ba? Medyo dako jud siya, no? Blot-like. Okay, ayan. So that's for the trough of Endolimax niina. Okay, all right. Next, we go to the next genus, okay, that again does not contain peripheral chromatin nor chromatoidal bodies, and that is Iodamoeba buwechlii or buchlii. So, kani siya ang spelling dears if namoy duha ka dot sa U. But if wala ni siya dot, dapat ang spelling ana kay B-U-E. Okay, B-U-E. So, murag, again, I think that's, German man ata ni, I'm not sure lang. Okay, so Iodamoeba buwechlii. Okay. Now, for the cyst, again, 10 to 12 micrometers ang size, round to oval. Very important characteristic, usa ra ka nucleus when mature. Okay, one nucleus ra gid siya. And large eccentric caryosome. Peripheral chromatin is absent. Quite same pod sa imuhang Endolimax niina. Okay, pero eccentric yung caryosome, dili blot-like. And coarsely granular sa cytoplasm. And very important feature sa cyst is the presence of a large iodine staining glycogen vacuole. So nasa pangalan na, 'di ba? Iodamoeba. So ioda, na siya iodine, ayan, iodine staining glycogen vacuole. So nasa pangalan na Iodamoeba, very important. Okay, okay. And of course, negosyo pretest contains mga achromatic refractile granules na pwedeng makitaan on one side of the caryosome, making the nucleus appear as if mura siyang basket or a basket of flowers. So murag appearance sa iyahang granules na nasa kilid sa caryosome kay murag flowers in a basket. Kay nagtapok man sila. Okay? So basket nucleus, basket of flowers na nucleus. Again, large iodine staining glycogen vacuole. Nako, that's all for Iodamoeba buwechlii. Okay? So of course, buchlii or buwechlii, iyahang translation is actually a basket, no? So nasa pangalan na Iodamoeba, iodine staining glycogen vacuole, buchlii basket. Ayan. So basket nucleus, iodine staining glycogen vacuole, all characteristics of Iodamoeba buchlii or buwechlii. So illustration of your cyst, ayan. So as you can see, very important, di ba? Large glycogen vacuole. Cytoplasm is still granular. And of course, you have here a basket appearance. No, marasag flowers in a basket. Kaya nagtapok ang imong achromatic granules or refractile granules. So here's an example of an iodine stained cyst, di ba? Very, very important or klaro imuhang glycogen vacuole na stain siya by iodine. Okay? But using your trichrome stain, of course, dili siya ma-stain. So it will remain colorless. Pero klaro kayo siya na structure. Okay? So this one, if makakita ka, no, dako kaayo na glycogen vacuole, then press the buzzer kay Iodamoeba buechliae na siya. Okay? Alright. Ayan. That's for Iodamoeba buchliae cyst. Okay, all right. Now for the trophozoite, medyo large siya, 8 to 20 micrometers in diameter, fairly active motility, granular cytoplasm pa rin, vacuolated, and could have a presence of a halo, no, around the nucleus because of during staining. And there could be again chromatin granules pa rin na pwedeng nagpalibot sa iyang caryosome. All right, so pwede dyan pong basket appearance or basket of flowers appearance. Okay, ayan. Now, this is an example of your illustration again. Vacuoles, granular cytoplasm. You have again no peripheral chromatin. Your chromatic granules kay nagpaligid or nag-surround na sa imuhang caryosome. Okay, and of course, here's a picture, di ba? You have here the cytoplasm, granular. You have the caryosome, di ba, large, and you have sa kilid, kabante mo ani na yung mga granules, na yung mga dots. Okay, so these are the achromatic granules, okay, that nagpalibot sa iyahang iyahang unsa ni caryosome. And still the same, di ba, kabantay mo na murag dili siya solid gani ang imuhang circle or ang nucleus because wala siya peripheral chromatin. Unlike sa imuhang Entamoeba histolytica, Entamoeba coli na yung mga peripheral chromatin. Okay? For your Iodamoeba buechliae and for Endolimax niina, you can see, di ba, medyo hollow ang sulod sa imong nucleus. Murag wala siya solid na color because again wala siya peripheral chromatin. Okay, now these achromatic granules again are quite difficult to see under preparations. Okay, so sometimes siya makitaan. Alright? So muna siya appearance sa imong Iodamoeba buechliae na trophozoite and could have a presence of a halo appearance during staining. Okay, ayan. So again, your Iodamoeba buechliae and Endolimax niina, actually they look the same, no? They have quite the same characteristics. Granular cytoplasm, no peripheral chromatin, di ba? Vacuolated in the cytoplasm. So how to differentiate? Again, you look at the different mga defining characteristics. Like for Iodamoeba, di ba? Sa cyst contains a large iodine staining, glycogen vacuole. For the trophozoite, you could see mga granules if naa, okay? And also look at yeah, the granular cytoplasm and the presence of a halo, di ba, during staining. All right. And for Endolimax niina, again, quite smaller, medyo oval iyahang cysts, all right? And the trophozoite, again, blot-like caryosome. All right? So again, medyo smaller po siya. All right? Ayan, that's for the intestinal amoeba nato na non-pathogenic. Okay. So before we proceed to Entamoeba gingivalis, so you may be asking, sir, like, why do we need to study about this? No, why do we need to talk about the non-pathogenic? Na di man sila makakosog disease, di ba? So why nga, di ba? So muna siya question nato. Do we report ba? Ayan, ako i-ask ninyo. Do we report ba? No, do we report these non-pathogenic intestinal amoeba when we see this in our stool samples? Ato ba silang i-report sa imuhang lab results? Yes or no? Sige. Ato ba silang i-report? Let's say nakakita kag sample na contains Entamoeba coli na cyst or Iodamoeba buechliae trophozoite. Do you report that in your laboratory results ba na Iodamoeba buechliae trophozoite seen, Entamoeba coli cyst seen? Do you report or dili ra? Ato ra siyang i-skip.
Report.
Report, okay. Final
Maybe.
answer?
Maybe, sir. Maybe.
Maybe. Okay, sige. All right, I'll take that. Sige, yun sana pala yung answer. Ato ba siyang i-report or ato ra siyang i-skip to site? Ato i-skip to site or i-report ba nato siya?
Skip.
Skip? Okay, sige. All right. Skip to site daw. Okay, ato siya i-skip to site. Okay. All right. So, sa man tong answer, Anif? Ato siya i-report or ato siya i-skip to site?
If it's non-pathogenic, sir,
okay,
dili na dagay.
okay.
Report.
Okay, so there's no a need. There's not a need. Okay, ano sa pa answer? Natin ni answer. Dunga ni Cleo. Yes, do you agree ni Cleo? Na dili ra? Or
Yes.
sa man? Okay. All right. Sige, sige. All right. So interesting answers, no? But the correct answer, not really correct answer, but the recommended, no, is you still have to report it. Okay. So that was a question of my CI na nag-orals me sa CM sa Galyares gyapon and wala ko katubag pod no. So I think same tag answer Clay and the rest na dili ra report because again ako mindset ato kay yeah, why do you have to report it na non-pathogenic man no? Like what man siya nag-cause of disease, so why do we have to report it? So but theoretically based sa Zybig and other books pod, we have to report the non-pathogenic intestinal amoeba even if they do not cause infection. Why? Because no, recall how these amoeba are transmitted to humans. It's through ingestion of fecally contaminated fecally contaminated food or water that contains the cysts. Okay. Now, the presence of these amoeba, bahalag non-pathogenic, in a patient sample could indicate that the patient's source of food or water is fecally contaminated. So, that's why through our reporting of this in the laboratory result, we could inform the patient or the doctor could have an idea and the patient will then be aware na the source of his or her food and or water is contaminated with feces. Ayan, kay nakakuha man siyang amoeba, okay? From the food or water na iyahang gikaon or based sa sample na itong nakita. Okay? So that's the importance of it. That's why we report it pa rin. So it's more on preventive measures for the patient because sooner or later no if magpadayon gyapon di nato i-report no na natin nakita na non-pathogenic amoeba. Now what if magpadayon gyapon siya sa source of food and water niya? So sooner or later makakuha na gyud siya sa pathogenic which is your Entamoeba histolytica because again di ba their mode of transmission is the same. It's still through ingestion of fecally contaminated food or water that contains the cysts. Okay, so that's the importance of reporting these non-pathogenic intestinal amoeba. So please take note of that rationale ha. Atong reason nga nandun i-report kay base ni pangutana sa inyong oral. Sala, yes, o di ba? At least. Pack, press the buzzer na. Plus points, plus, plus, plus. Perfect. Okay? Alright, ayan. So that's the importance. No? So bahalag non-pathogenic, bahalag for you, dili siya important. No? Ato gyapon siya ngatagan og importance. Ato siyang i-report kay dili lalim na dili mahatagan og importance. Charot. Okay, drama. Okay, ayan. Sige. That's for the non-pathogenic intestinal amoeba. Okay? All right. Now we go now to the last no non-pathogenic amoeba, pero di lang siya intestinal. Your Entamoeba gingivalis. Okay, now Entamoeba gingivalis was considered to be the first amoeba described by humans, no? And it was found from the teeth, of course, or from the mouth, Entamoeba gingivalis. Now, this amoeba is the only one that ingests WBC. So, very defining characteristic sa imong Entamoeba gingivalis na ay WBCs sa iyahang cytoplasm. Now, another thing, wala siya cyst. Okay? Wala siya cyst, pero na siya bro, charot. Okay, na siya. Wala siya cyst, but of course, na siya trophozoite diet. So, na siya trophozoite. The trophozoite, 5 to 20 micrometers, active motility, single nucleus containing centrally located karyosome, and finely granular cytoplasm, and a very important inclusion is WBCs. The trophozoite can also ingest RBCs, but it's not common. Okay? Mas common dyan po ng E. histolytica na mo ingest of RBCs. Okay? So your E. gingivalis, WBCs ang pinaka-important na inclusion sa iyahang cytoplasm. Okay, so here's an example. Illustration, ingested WBC. And in picture, stained niya siya using Papa Nicolaus, I believe. All right. So kaning gi-point na arrow that is the WBC na gi-ingest. Okay. All right. Ayan. So another picture, other pictures you have here again. Ayan. So again, this is the ingested WBC. Okay, and this is the pseudopod. So if you can see, no, the characteristics of the trophozoite is quite similar with the characteristics of your Entamoeba histolytica din na trophozoite. So it's important that we differentiate the two because in cases of pulmonary abscess, no, pulmonary amoebic abscess, pwede natong ma-recover ang E-hist or E. gingivalis at the same time. Because again, di ba, pulmonary, so sa sputum sa patient, there could be, again, E-hist or E. gingivalis. So how do we differentiate is again through ingestion, through the cytoplasmic inclusion. So we look for WBCs or RBCs. So kung WBCs atong nakita, that's Entamoeba gingivalis. Kung RBCs atong nakita, that's Entamoeba histolytica. Okay, so quite difficult din naman to look into, no, basta kailangan mas important siya na i-stain nato siya, no, stain smears, because that would really help us in looking at the cytoplasmic inclusions. All right, so if WBC ang sulod, E. gingivalis, but if it's RBCs, E. histolytica. All right, so generally Entamoeba gingivalis can be recovered from the mouth, okay, but there have been cases na na-recover siya from the vagina. Okay, so I don't know how it got there. ask me ha. It's not what you think then, and it's not my cup of tea. Charot, okay, ayan. So, Entamoeba gingivalis was recovered in some patients, no, from the vagina. And it was found out that it came, or yeah, it came from patients who used intrauterine devices, mga IUDs, no? And IUDs are contraceptives, no? So, kato mga patients na naggamit og contraception na IUD, na po yung mga nakuha na cases of Entamoeba gingivalis. Pero wala po sila nag-exhibit og symptoms. So, it still remained non-pathogenic in the vagina, pero na-recover lang siya. Okay, so again, I don't know how it reached your IUDs. Okay, could be na-contaminate siya or I don't know. Okay, basta na-recover siya. The fact lang is na-recover siya from the vagina. Okay, okay, ayan. All right, okay, that's for Entamoeba gingivalis. But generally commensal siya of your mouth. Okay, ayan, sige. So that's for Entamoeba gingivalis. Okay. All right, so before we continue to the last type of your amoeba, the free-living amoeba, do you have any questions, dears, before we continue? No? Si E. gingivalis, nabot na siya sa vagina. Oh, so- Grabe naman to si E. gingivalis. Okay. Ayan, any questions? All right. So, kanina siya na amoeba, again, exhibits nuclear variation, can mimic D. fragilis or Entamoeba Heartmanite. Kinsa ni siya?
Endolimax, sir.
Okay, Endolimax. Sa tong ano?
Niina.
Species.
Niina.
Okay, very good. Endolimax Niina. Okay, next, this is considered to be the small race of Entamoeba histolytica.
E. Heartmanite.
Heartmanite.
Okay, very good. Okay. Again, this is the most common amoeba in in Papua New Guinea, parasite of pigs and monkeys.
Poleki.
Okay, all right.
Lecky.
Entamoeba poleki. All right, very good. And this contains a large iodine-staining glycogen vacuole in the cysts.
Amoeba
Okay.
Butchley.
Okay, perfect. Amoeba buechliae or buchliae. Okay, perfect. Very good. And of course, the only amoeba that ingests WBCs and the first amoeba to be
Entamoeba
discovered.
gingivalis.
Gingivalis in the vagina.
Again, Entamoeba gingivalis. Okay, it was recovered in some patients, no, from the vagina, but not common din naman. Okay, there have been cases. Okay, there have been reports. All right, ayan. So that's for the non-pathogenic amoeba, both intestinal and Entamoeba gingivalis. Okay, all right. So we'll now proceed to the last type of your amoeba, your free-living amoeba. Okay, now free-living amoeba or opportunistic amoebae. Okay, amoebae. All right. So this consists of your Naegleria, Acanthamoeba, and Balamuthia. Now, Acanthamoeba and Balamuthia, silang duha gyud ang mo exhibit or makakause of cutaneous lesions. Okay, or cutaneous infections in humans. For Naegleria, usually dili siya mo cause of cutaneous infections. Now again, when free living, okay, in the environment, no, it does not cause disease. Mo live ra siya in harmony with the environment, mag swim sa water, sa soil, mukhaon og mga bacteria and all that, no, very high ang life, no. But once it reaches the brain or once it reaches humans, it now causes infections. Okay, all right. So same siya with strongyloides na it could be parasitic or not, no. So facultative parasite, it can live with or without the host. Okay, pero siya sa environment, no, non-parasitic, or when it becomes or when it is transmitted to humans, they now become pathogenic. Okay, all right. Now, there have been bacteria, very important, si Legionella pneumophila, that can reside, no, can infect, multiply within, and kill your Naegleria and Acanthamoeba amoebae. So, mara siya in a way, gi-hijack niyang amoeba or ni parasite po siya, ni parasitize po siya sa imuhang amoeba, free-living amoeba. So, aside from Legionella, you could also have these organisms, Campylobacter, Listeria, Vibrio, Mycobacterium, Burkholderia, and Pseudomonas. So, mga bacilli usually. Okay? All right. Now, because of the presence of these bacteria in some amoeba, no, pwedeng mo aggravate, pwede siya mo contribute kanina mga bacteria sa severity sa infection caused by the amoeba. Okay? So, na possible contribution, no? Na contribution, sana nagdala ko ng pansit, charot. Okay, so again, contribution sa bacteria that lives within the amoeba to the infection caused by the amoeba. Okay? Ayan. So, Legionella pneumophila, okay, Legionella, unsa ganito ang sorry. Unsa tong media of choice for Legionella pneumophila? For isolation of your Legionella pneumophila. Sa man to na agar or culture media.
Buffered charcoal yeast extract, sir.
Okay, all right, very good. So your BCYE. Don't forget the culture media of choice for Legionella pneumophila, your buffered charcoal yeast extract agar, your BCYE. Very good. Okay, ayan. So don't forget about that. Media of choice for Legionella pneumophila. Okay, all right. So that's for the introduction to our opportunists or your free living amoeba. All right, so we'll now start with the first free living amoeba, your Naegleria fowleri. Now again, Naegleria fowleri is the causative agent of your PAM. So nagawa sa imuhang pretest, the major causative agent of your primary amoebic meningoencephalitis is Naegleria fowleri. Okay, and it's considered to be your brain-eating amoeba. So siya may primary, siya ang cause of primary amoebic meningoencephalitis. Kinsa man day cause sa secondary amoebic meningoencephalitis? Kinsa may nag-cause ato sa secondary amoebic meningoencephalitis?
Entamoeba histolytica.
Okay, all right, very good. So ang Entamoeba histolytica, once it reaches the brain, it now causes your secondary amoebic meningoencephalitis. Okay, all right. But for PAM, for primary amoebic meningoencephalitis, it's Naegleria fowleri. Again, yung colloquial term, no, or is also well known as your brain-eating amoeba. Okay, so sa mga news articles, usually gina-refer siya as the brain-eating amoeba. Okay, so kung si Naegleria fowleri mong brain-eating amoeba, kinsa man tong flesh-eating bacteria?
Group A strep.
Okay, all right, very good. Your group A strep. Okay, kinsa man ang species sa group A strep?
Pyogenes, sir.
Pyogenes ba? Dili pa si
Ay,
Agalacti.
Di, Pyogenes, sir.
Pyogenes or Agalacti?
Pyogenes.
Sure, dili ba si Pneumoni po?
Dili,
Dili,
sir.
sir. Dito, sir.
No.
B si Pneumoni?
D.
Agalactiae ang B.
Okay, nya si Pneumoni, unsa man siya na group?
D.
D? Si Pneumoni?
Wala.
Wala?
Mansfield. Yan.
Okay, all right. Okay, ayan. So, question na mo, dapat, para namoy convictions yung answer. So, that's true. Your group A strep is strep pyogenes. Your strep pneumo, ha, wala siya Lancefield antigen, so wala siya group. Okay, di ba? Very important. Ayan, sige. So, very good. Your flesh-eating bacteria, strep pyogenes. Brain-eating amoeba, Naegleria fowleri. So, kana mga other names. Okay, all right. So, very good. Again, this is the major causative agent of your PAM, primary amoebic meningoencephalitis. Now we go now to the mode of transmission. How do we get this free-living amoeba? It's through inhalation or aspiration of water. Okay, primarily water that contains your trophozoites. Trophozoites good ang ato ang infective stage. Okay, now the infective stage, again, trophozoites, siya yung musulod sa imuhang nose. Okay, so through inhalation. Usually inhalation of water during, ayan, sa mga mahilig mag-swim dyan, mga swimming. Okay, ayan. So again, through swimming, usually, no, pag swim, maka-inhale ka sa water that contains a trophozoite of amoeba, dito na siya makakosog infection. Okay? Alright. Now, primarily, kani siya na type of amoeba. Okay? The amoeboid na forms imuhang Naegleria fowleri. Now, once it reaches, again, the nose, it now makes its way padulong sa brain where it causes infection. Okay, so it's primarily through swimming, okay? Swimming sa mga unknown bodies of water. So sa mga mahilig mga mag-vacation, yes, okay? Mga swimming, no? Especially in mga waters na medyo init, no? During mga warmer climate or warmer seasons or mga unknown bodies of water like lakes, no? Mga hot springs, mga streams, okay? Because dito ganahan si Naegleria fowleri. Ganahan siya init, no? Ganahan siya magpainit, charot. Ganahan siya init, thermophilic siya. Okay? So if you like to go swimming, mga unknown bodies of water, ba kanang murag mga bodies of water na makain ka na murag sharks, maraming naay ka po yung aning charot kanina na gani na scary. So dito na mga bodies of water, especially mga init, okay? Or during mga warmer seasons, warm ang weather. Okay? So mo proliferate kaayo si Naegleria ana. So sa swimming, okay, make sure lang that your head is above water, okay? Na dili masudlan og water mong ilong. Okay, because that's the mode of transmission of Naegleria. Okay? All right. Ayan. So inhalation or aspiration of water. Now, another species that is of interest, no, that could infect humans down in the future is Naegleria australiensis, but it's still being studied, no? So it's still not defined or it's not established yet that can cause infection. Okay, so ang primary species pa good is Naegleria fowleri. Okay, all right. Now we go now to its morphology. So for the trophozoites, very interesting for Naegleria fowleri and very unique is that it can exhibit two types of trophozoites. One is an amoeboid and the other is a flagellate. And the amoeboid form is what we see in humans and also the infective stage to humans. All right. Then the flagellate form can be found in the environment or during culture put. It can also be found in tissue in humans pero not common. Seldom lang, okay? So ang pinaka-common good is the amoeboid form of trophozoite. And because it has the ability to convert from amoeboid to flagellate, it's also known as an amoeboflagellate. Okay? So duha ka forms of trophozoites ang naa niya. Okay, so the first form is the amoeboid form. Amoeboid, medyo dako siya sa front and then medyo tapered or gamay siya sa end, posterior end. Large central karyosome, no peripheral chromatin, granular cytoplasm that contains vacuose and slug-like blunt pseudopods. And this one, I think wala na ako nabutang, trailing effect in your agar plates with gram-neck bacilli. So trailing effect meaning na yung mga movement on the surface of the agar, okay? Because nag-move siya within the agar, okay? On the surface of the agar, okay? So trailing effect on agar plates that contain gram-neck bacilli. So here's an example of your amoeboid na trophozoite. So again, blunt pseudopods, as you can see, dili sharp, no, ang ends ng pseudopods. Granular, vacuolated ang cytoplasm, and very important, spherical nucleus with a large karyosome. So that is the most prominent feature natong lantawon for Naegleria fowleri. Dapat naa siya spherical nucleus, spherical nucleus with a large central karyosome. Okay, spherical nucleus with a large central karyosome, defining characteristic, prominent characteristic of Naegleria fowleri. Okay, all right. So that's for the amoeboid na form. Okay. Now again, another type of trophozoite niya is the flagellate. Of course, the flagellate occurs when the trophozoites are transferred from culture or sa tissue mangani, nagibutang og water. Okay. Now the flagellate again contains two flagella and it has a jerky, no, magkurog-kurog siya, or spinning motility. It can withstand moderate heat, but it dies in chlorination or high salinity. Okay? All right. Ayan. Now, this is an example of the flagellate. Again, duha ka flagella. All right, and medyo pear-shaped iyahang appearance. Again, the flagellate form can be seldom lang ma-recover sa humans, okay? And usually makita siya sa environment. And in the environment, the flagellate form of your amoeba or the flagellate form of your Naegleria fowleri assists in the transfer of Naegleria fowleri from one body of water to another or from one environment to another. Because again, flagellate, mas paspas siyang makalihok. So mas paspas siyang makaadto to different places in the environment. So pwede siyang maka-transfer from one body of water to another. Okay, so sa flagellate form. All right. So once the flagellate form, example, flagellate form nakuha sa water, mo convert siya to amoeboid. Ayan. So inana siya ka, you know, flexible, okay, in a way. It can convert to amoeboid. Example, kung flagellate mo nakuha, no, pwede siyang convert to amoeboid and then dito na siya mo cause infection. Okay? All right, ayan. But generally, again, the trophozoites, dears, are again prone, no, are sensitive to mga chlorination. That is why it's important na to mga tap water, no, atong sources of water are chlorinated. So sa US usually common ni siya because na some states na ilahang water is not chlorinated enough. Okay, so makasurvive ang trophozoite. But if ang water that contains trophozoites mainom nimo siya, okay, it will not cause infection because that's not the mode of transmission man sa imong Naegleria. But in cases lang na example kanang maligo ka, no, masudan nimong ilong, maka aspirate kag water sa imong ilong that may contain, no, the trophozoite of Naegleria, then dito na siya maka-cause infection. But if drinking lang siya, less likely because again that's not the mode of transmission sa imong amoeba, okay, sa imong Naegleria fowleri. All right, that's for the trophozoites. Okay, two ka form of trophozoites, duha ka type of trophozoites. Now for the cysts, ayan, so single nucleus, again not present in tissue. So dili jud nato makitaan siya sa human tissue, sa brain, sa CSF ang imuhang cysts. Okay, all right. Now, it has a smooth double wall, and of course, because it has a double wall, strong ang structure, it can resist unfavorable conditions. Okay, and that is, di ba, the main purpose of your cysts, bahala kinsa pa na amoeba, no, or bahala kinsa pa na protozoa, they are usually formed during mga unfavorable conditions, harsh environment, because again, to protect the organism. from dying. Okay. All right. So here is the cyst, no, of your Naegleria fowleri. Double wall, di ba? Strong iyahang structure, and you have a single nucleus. Okay. And again, the cysts cannot be seen in your tissue, in brain tissue or CSF ba. Okay. All right. So we go now to the disease that it causes. Again, it causes primary amoebic meningoencephalitis, or your PAM. Now your PAM, primary amoebic meningoencephalitis, is described as acute suppurative. So meaning possible na yung mga pus na mga ma-produce or mga abscess. Okay? And in humans, usually PAM is rapidly fatal. Okay? Bahalag matreat pa siya. No? More than 95% of the cases, even if treated, no, they lead to death. Okay, so because prognosis sa imuhang PAM is bad. Okay, it's not good. All right, so it's rapidly fatal in humans. Okay, now your N. fowleri, it directly ingests yung ginakaon mong brain tissue through the process of amoebostomes, no, or food cups, no. So amoebostomes. So these are structures sa yung cytoplasm na murag mouth niya. So dira siya mo gamit. Muna yung gamiton para makakaon siya sa brain, no? And aside from that, it also lyses the cells of the brain. Okay? So that's why it's deadly. And paspas pag yun iyahang disease progression. Okay, all right. Now, it can resemble acute bacterial meningitis at the early stages. So that's why most of the time it is misdiagnosed as bacterial meningitis, because ang presentation sa yung symptoms and laboratory results po sa CSF or whatever, they are consistent with bacterial meningitis. So the doctor can misdiagnose it as bacterial meningitis and give treatment to bacterial meningitis such as antibiotics, which are again not effective against your amoeba. So it could lead to misdiagnosis at the early stage. So dili noon matambalan ang real disease. So mas mo aggravate, mas madugay ang diagnosis and treatment. So mas mo contribute sa pagkapaspas sa disease progression. Okay, so again, that's why it's also fatal. All right, ayan. So hemorrhagic necrosis, usually after autopsy makitaan na there is hemorrhagic necrosis, nangamatay, no, na bleeding sa mga olfactory bulbs. Of course, dito nag-start, no, ang imuhang amoeba pagsulod sa nose, and then iyahang gipangkaon to mga cells dito, and then it made its way padulong sa brain. And at the base of the brain po na yung mga hemorrhagic necrosis or bleeding and mga dead cells. Okay, all right. And again, this one, dears, I think wala na ako na-appeal. The patients also show Kernig sign, which is a diagnostic sign for meningitis. Okay, so if imong ipa-flex ang hip sa patient, dili po niya ay if imong ipa-flex ang leg or imong ipa-straighten ang leg sa patient while naka-flex iyang hips, dili niya mahimo, because there could have already been destruction sa mga nerves, no, between the brain and the muscles dito. So munang dili niya mahimo na. Okay, so diagnostic sign of meningitis. Okay, ayan. So that's for PAM. Again, very acute, no, very fast ang disease progression. So within 3 to 6 days, no, after exposure to the patient, or within 3 to 6 days after symptoms, ayan. Within 3 to 6 days after onset of symptoms, the patient can die, no, or the patient could lead to death. Okay, ayan. So ina siya kapaspas 3 to 6 days, no. So kailangan paspas pyud ang diagnosis and treatment. But again, even if with treatment, more than 95% of the cases of PAM has bad prognosis or leads to death. Okay, ayan. All right. Now for this is your amoebostomes. Again, kani mga circles. These are the food cups or food suckers of the trophozoites of your amoeba, no? So this is what your amoeba use as a mouth, no, para makakaon siya of different brain tissue or cells na iyahang giagian. Okay, all right, ayan. Sige. Now we go now to diagnosis. So how do we diagnose? Is of course through CSF examination or brain biopsy ba ng mga specimens. So what we look for is again sa CSF or sedimented CSF, we look for motile trophozoites. So we add 1 drop of CSF on a slide, put a cover slip, examine under the microscope, and look for motile trophozoites of CSF, mga amoeboid. Or if ato siyang gi-concentrate, sediment, no, we then make a smear and then stain with your blood stains, rights, or GEMSA. Now for centrifugation, again, it should be done at a low speed, 250 times G, and should never be refrigerated. Why? Because this could destroy, no, or this will protect your trophozoites na dili siya ma-destroy. Okay, now sa atong discussion sa CSF samples sa mga diagnostics katong first lecture nato, di ba? Medyo paspas ang speed dito sa CSF. So it would depend on the organism that you want to recover sa kapaspason sa imong centrifugation. So for Naegleria foliate na trophozoite, again, slow speed lang, 250 times G. And of course, never refrigerated because it could destroy your trophozoites. Okay? All right. Now for CSF analysis, no, 'di ba? Recall in your AUBF, 'di ba? We perform CSF testing, 'di ba? Tulo ka test tubes atong isend sa chem, 'di ba? Sa hema and of course sa micro. So ato na siyang i-test. For chem, ayahang results is of course decreased glucose concentration, increased protein concentrations. And for cell count, increased imuhang PMNs, no, leukocytes, from several hundred to greater than 20,000 cells per millimeter cube. Now, di ba, kanina pa lang daan na results, usually sila may unang mahuman after og test sa CSF, di ba? So pag release ani na results sa doctor, of course, the doctor can interpret that as consistent with bacterial meningitis, di ba? So, dira pa lang daan, tambalan na siya for bacterial meningitis. But then, again, that's not the cause of the meningitis. It's caused by Naegleria fowleri, di ba? So that's why pwede siya ma-misdiagnose as bacterial meningitis in the early stages. And after culture, mugos ng culture, dira ka makakita na negative yung gram stain and negative yung bacterial culture. So dira pa makaingon ng doctor na, ay shucks, dili day siya bacterial meningitis, shit, ganong level. So kailangan mag-change og treatment, ganun. But again, if the doctor also looked at the history of the patient, no, na history of swimming, history of contamination with water ba, then the doctor can also point na, no, that basin possible na PAM ni siya, even without the result of the culture or gram stain. Okay, because again, laboratory results, there's radiological tests, these are just supplementary, okay, to the initial diagnosis of the doctor. Okay, sa history pa lang daan, majority na daw of the diseases can be diagnosed. Okay, so sa history pa lang daan, it can already give the doctors, no, a brief background or a glimpse or an initial presumptive diagnosis as to what the patient is experiencing. Okay? Ayan. So, supplementary na lang na siya. Alright? So, but ang point lang is based on mga chemical analysis sa CSF, same good siya results with bacterial meningitis. That's why pwede siya ma-misdiagnose. Okay? Alright. So, CSF analysis, ayan, AUBF, last subject na to for didactics, no, in the later part of your first semester. So, CSF, again, recall, kinsa man tong type of meningitis na maka-cause or na ay appearance na pellicle formation sa imuhang CSF after overnight incubation at room temperature. Na ay pellicle formation. Na yung appearance of pellicle. Tapos sa man to na sakit. Remember pa ba itong AUBF dyan? Okay, alright. Pellicle formation sa imong CSF, kinsa to na type of meningitis. Bacterial ba?
Tubercular, sir.
Tubercular. Okay. Napailang answers? Okay. Alright. Tubercular ba? Tama ba yung may pellicle formation? Tubercular ba? Alright. Okay, so I believe wala na mo answer. And that is the correct answer. Yes, very good. So tubercular meningitis, if yung MTB mabot sa brain, can cause meningitis or sa mong meninges, no? Ihang CSF appearance can ay pellicle formation. Okay, pellicle formation in the CSF, tubercular meningitis. Okay, now gram staining. Ayan. Iyahang appearance sa gram stain sa CSF, starburst appearance. No, starburst appearance of gram stain in your CSF kinsa man yung organism.
Cryptococcus neoformans, sir.
Okay, all right. Very good, very good. Ang galing. So, Cryptococcus neoformans. Okay, Cryptococcus neoformans, kung ato siya i-gram stain, ang iyahang appearance sa CSF is starburst. Mura siya daghang stars, no? Starburst. Okay, dili tong restaurant ha. Starburst appearance. Okay? Ay sunburst dito. Shucks, Mark. Okay, kaulaw. Starburst appearance gram stain is again Cryptococcus neoformans. Pero kinsa nitong unsa tong stain of choice for Cryptococcus neoformans gani? Ma-remember pa ba? Unsa to na stain atong gamiton?
India ink.
Okay, very good. India ink. Galing, galing. Okay, that's good. So India ink atong stain of choice for Cryptococcus neoformans, and it also has a starburst appearance in CSF na gram staining. Okay, perfect. Galing, galing. Please keep it up, dears. Okay? Okay, padayon lang sa pag-review and pag-recall. All right, okay. So again, that's for CF analysis sa PAM. So very consistent with bacterial meningitis. Okay? All right, ayan. Sige. Now we continue for culture. Now if you want to culture your Naegleria, pwede po. You can use non-nutrient agar as long as it contains E. coli because E. coli, Escherichia coli, will now serve as the food source of your amoeba. Now kaning incubation dire dears, wala na ako nabutang, no? 35 to 37 degrees Celsius kung gikan sa CSF or gikan sa brain ang sample and 30 degrees Celsius if gikan from other tissues. Okay, all right. Now, histological preparations, mga brain biopsy, no? We use indirect immunofluorescence assays or immunoperoxidase techniques, okay? Now, understanding it may look like Iodamoeba boettchleii trophozoite because recall, di ba, your Naegleria foliae trophozoite, it contains, di ba, a large caryosome and a spherical nucleus, which is similar with the Iodamoeba boettchleii, di ba? I. boettchleii contains a large caryosome and spherical nucleus pag yun. But look at the specimen kung asa siya gikan. It's from brain or CSF. And your Iodamoeba boettchleii dili maabot sa CSF or sa brain. Okay? So for histological preparations examination, medyo advanced ang techniques that you need to perform, immunofluorescence or immunoperoxidase. So again, not routinely performed usually sa imuhang hospitals, na routine hospitals. Pero mga high-end hospitals, yes, pwede ni siya. Okay, alright, ayan. Sige. Next, if N. fowleri atong cause, so CNS infection, ang makita na nato is again trophozoites sa tissue, whereas for Acanthamoeba, pwedeng cyst and trophozoites. Ayan, that's the difference. So if ang Naegleria, ato ang causative agent sa CNS infection, trophozoites atong makita. But for Acanthamoeba, if siya ang cause of infection, pwedeng cyst and trophozoites atong makita sa tissue. Okay? Alright. Also for cultured ideas, sa imuhang Naegleria fowleri, don't forget, it cannot survive if ang concentration sa salt, NaCl, is greater than 0.4%. So if medyo high imuhang salinity or high imuhang salt concentration, dili na makasurvive imuhang Naegleria fowleri. Okay? Alright, ayan. So please take note of that. So that's for Naegleria fowleri. So here are some pictures. So this is from brain tissue, again, of Naegleria fowleri, infected na patient, no? So again, large caryosome, spherical nucleus, okay? And here's an example of Naegleria fowleri trophozoite in a cytospin or gicentrifuge na CSF, di ba? As you can see, daghang PMNs, neutrophils, and the trophozoite, central caryosome, no? And you have a large nucleus, spherical nucleus, okay? All right, so again, daghang PMNs. All right, so that is, again, your N. fowleri trophozoite. Again, pinaka-common form na ma-recognize nato in humans is the amoeboid trophozoite. All right. So again, that's for Naegleria fowleri. Causative agent of your PAM, primary amoebic meningoencephalitis. Brain-eating amoeba. Okay. Mode of transmission is inhalation, aspiration of the organisms, especially during swimming. Infective stage is the amoeboid trophozoite. Okay. But it contains three forms. You have amoeboid trophozoite, flagellate na trophozoite, diba? And you have also the cyst. Okay. Cysts can never be seen in tissue. Trophozoite try makita. Okay. All right, ayan. So that's for Naegleria fowleri, our first free-living amoeba. All right, so before we proceed to the next, your Acanthamoeba, do you have any questions, dears? Okay, so be careful ha if mag-outing nyo sa mga friends, yung mga blockmates, no, kanang magligo-ligo mo. No, sa dagat naman, hindi naman siya medyo makita dito, but for fresh water usually, no, kana mga somewhat na warm freshwater mga lakes no may nana, so be careful. Right, but in the Philippines di kaya siya common, no, but in other countries, especially sa US, very common siya dito. Daghang mga cases dito of Naegleria fowleri infection. And again, most of the time, majority, no, more than 95% of the cases of PAM leads to death, no, so very very deadly gyud siya. Okay, so that's for Naegleria fowleri, the brain-eating amoeba. Okay. And kisa itong flesh-eating bacteria na to?
Streptococcus
Streptococcus
pyogenes.
pyogenes.
Okay, strep pyo. Okay, your group A, streptococcus. Kinsa na itong causative agents atong erysipeloid? Hala, kalimtan na. Kinsa itong causative agent nato sa erysipeloid? Si group A strep pa ba Japon? Erysipeloid. Hala.
Rucipathiase.
Okay. Si Erysipelotrix.
Erysipelotrix.
Okay, Erysipelotrix, Erysipelotrix rujopathy. Diba? I told you how to differentiate Erysipeloid, si Erysipelotrix. Kung Erysipelas, diba, AS ang end, caused siya by group A strep. Okay, don't forget that. All right. Okay, so that's all for Naegleria fowleri. Okay, so we now continue to the next free living amoeba, and that is your Acanthamoeba species. Now, Acanthamoeba species, because it's Acanthamoeba species, kay daghang species na pwede maka-infect sa man, no? So, muna sila. But ang pinaka-common is si Culbertsoni, okay? And si Castellani. Si Castellani ang majority ang cause sa mga CNS and eye infections in humans. Okay? So si Culbertsoni and Castellani. But these species, again, can infect humans. That's why Acanthamoeba species atong ngalan kay daghan silang pwede. Alright? Okay, ayan. Now, different siya from Naegleria in that wala sila flagellated stage. Okay? So normal ra siya na amoeboid na trophozoite and a cyst. Okay? So wala siya flagellated na stage. And again, nasa pangalan na Acanthamoeba, iyahang pseudopods sa iyahang trophozoite contains spine-like pseudopods. Okay? And that is known as your Acanthopodia. Right, na-typo ko sa inyong notes there, na Acanthopodia na siya. So please change lang to Acanthopodia. Diba? So acantho, diba, from prefix na acantho, it refers to na yung mga spine-like protrusion sa cytoplasm. Diba? In your RBC, sa hema, acanthocytes. Diba? Acanthocytes, diba, characteristic of your A-beta lipoproteinemia na sakit. Yes, mga RBC morphology na to na abnormal. Please take note of that, dears. Yes, and mga diseases na related. Hema 1, yes, laban tayo sa hema. Okay, all right. Now, same here with Acanthamoeba, Acanthopodia, no? Spine-like pseudopods. Okay, all right, ayan. Now, for the modes of transmission, quite different din siya from Naegleria. But it's the same in the sense na you can still acquire it from aspiration or inhalation, okay? But inside of sa nose, okay, dito siya sa lower respiratory tract magikan or ulcers of the skin, okay? So mga samad sa skin. Now from there, it will now spread via blood, hematogenous spread, padulong sa imuhang brain, which will now lead to a CNS infection. Okay? So different siya from Naegleria in that gikan sa nose, gikan sa nose imong Naegleria padulong sa brain. For Acanthamoeba, gikan siya sa imong lower respiratory tract or ulcers sa imong skin. And giyon sa niya pag-spread, hematogenous via blood. Okay? And another mode is, of course, invasion of the eye, which leads to keratitis. Okay? All right, ayan. So here is your mode of transmission. Your Acanthamoeba and Balamuthia mandularis, same silang mode of transmission. Again, through lower respiratory tract, ulcers of the skin, spread via blood or hematogenous spread, okay, and leads to CNS infection. Okay, all right, ayan. So that's for your Acanthamoeba species. Okay, now we go now to morphology, starting first with your trophozoites. Again, characteristic, spine-like pseudopods or your Acanthopodia. Nasa pangalan na, di ba? Acanthamoeba contains Acanthopodia, your spine-like pseudopods. Okay. Now, the nucleus contains, again, daghang mga organelles, no? Large caryosome, organelles, Golgi complex, etc., etc. Okay, so here's an example of your trophozoites Acanthamoeba. Diba, you have a nucleus, no? Large caryosome, spine-like pseudopods, okay, spine-like pseudopods, and you have here again vacuose and the different organelles sa cytoplasm, okay? So ang distinct yun na characteristic or feature is the spine-like pseudopods. That's why it's called Acanthamoeba because it contains Acanthopodia. All right, okay, ayan, that's for the trophozoite. Next, another important thing is the cysts. Ayan, the cysts, general shape niya is round. Excuse me, it contains a single nucleus. And kani, very important, nigawa sa boards, not during our time, pero previous. It was asked, this parasite or this organism contains a double wall with an outer, ayan, outer wrinkled cyst wall. Ayan, dito palang daan. Outer wrinkled, okay, outer wrinkled, and an inner polyhedral cyst wall. Press the buzzer, atong answer na is Acanthamoeba. Basta double wall daw, and then ang giingon is, ang outer wall kay wrinkled, okay? All right, then that's Acanthamoeba species na cyst. Okay, and the inner wall sa imong cyst kay polyhedral, meaning pwede siyang different shapes, no? Pwedeng polygon, mga pentagon, ganun. All right, ayan. So that's for the cyst. Very important ha, take note, nigawa sa boards, not during our time. Okay, now cyst formation, again, like the other amoeba, it happens during unfavorable or harsh environment, no? And because the cysts, again, are strong structures, it can resist biocides, chlorination, and antibiotics, and even low temperatures. So here's an example of your cyst, diba? Generally round iyahang shape, pero iyahang outer, again, outer wrinkled, diba? Wrinkled kumot, okay, wrinkled outer cyst wall, and the inner is polyhedral. So polyhedral meaning mura siya it assumes a polygon na shape, 'di ba? This is a pentagon, 'di ba? In this type of cyst. All right? Pero ang pinaka-important, wrinkled outer cyst wall. Yeah, double wall siya, double wall. You have the outer and the inner. Okay? All right. Ayan. Sige. So that's for the cyst. Okay? Negwasa boards, please take note ha. Ayan. Basta ganit, double wall ha, double wall, wrinkled outer cyst. Nako, press the buzzer na wag na mag-isip. Entamoeba species. Diretso na tayo shade sa inyong answer sheet. Okay? And top notcher na. Charot. Okay. Amen. All right. Ayan. So that's for the cysts of Entamoeba. Okay, now we go now to the disease, no? These diseases, the siyang diseases na pwede makos. And the first of that is, of course, granulomatous amoebic encephalitis. So siya ang makakos sa GAE once it reaches your brain. Now, your GAE is usually pinaka-common with among no immunocompromised, immunosuppressed patients, or those that have already underlying condition for the longest time, chronically ill or debilitated. Those that are bedridden, no, those that are really kanang yeah, bedridden na, dili nakatindog and all that. So that's the difference, first difference niya from PAM. Your PAM is not only affecting immunocompromised individuals. Because sa PAM, bahalag healthy pa ka and your immune system is working well, once the Naegleria fowleri enters your brain, it can really lead to PAM. Bahalag intact pa yung immune system and you're living healthy. Okay, that's why your Naegleria fowleri is considered to be a true pathogen. It's not an opportunistic pathogen. True pathogen yun imuhang Naegleria fowleri. Wala siyang pinipili, okay? Bahalag immunocompetent ka or immunocompromised, no? The effects are still the same. No, it can lead to death and intense pud ang iyahang symptoms and effects. Okay? Now for GAE, it's primarily found in immunocompromised or mga immunosuppressed. Those that their immune system are not functioning well. Okay, all right. Now, unlike PAM, again, trophozoites and cysts can be seen, diba, in tissue. All right. And dissemination can also happen even in, especially in immunocompromised individuals. It can disseminate to other organs. And this type of disease or meningitis is more chronic, no? Medyo hinay siya ang iyang pag-progress sa disease, all right? Di siya pareha sa imuhang PAM na paspas. Now, within 3 to 6 days after onset of symptoms, pwede na mamatay ang patient. For GAE, pwede siya mo persist for a longer period of time. Okay, all right, ayan. Now, there may also be edema pa rin, hemorrhagic necrosis within the lobes of the brain, okay? And there are granulomas formed, and the blood vessels of the brain are thrombosed. So granulomas are parang mga kumpol-kumpol, clumps of cells, no? Pwedeng that are clumps of cells like immune cells, mga dead brain cells, whatever, because of the organism sa brain, okay? And thrombosed vessels, nihubag imong vessels sa brain, okay? There could be clots within, no? Because again, as a result of the organism, all right? Ayan. But again, bahalag progressive, chronic siya na form of meningitis. It can also lead to death pa rin. Bad pa rin ang prognosis sa imong GAE, okay? Or granulomatous amoebic encephalitis. Okay, all right. So here is some picture, your brain and GAE. So kaning mga red here is your, again, granulomas, all right, that may contain a lot of cells, mga immune cells, brain cells, no, etc. And as you can see, the blood vessels are thrombosed, meaning medyo nigahi siya or medyo enlarged, okay, as a result, again, of the organism. Okay, and here is an example of the cyst in brain tissue. Again, wrinkled outer cyst, and the inner is polyhedral. Okay, so again, cysts can be seen in tissue. Unlike sa imong Naegleria, na trophozoites lang ang makita sa tissue. Wala yung cyst. Okay, all right. So that's for GAE, or granulomatous amoebic encephalitis. So once the Entamoeba reaches the brain, muna siya iyahang disease na makos. Okay, all right. Next type of disease is, of course, Entamoeba keratitis and corneal ulceration. I think the more common type of disease na iyang ginakos. Okay, now this happens, of course, during, could be trauma, no? Example, ma-expose imong eyes through contaminated materials like maigo, no? Accidentally imuhang mata with a contaminated material that contains the Entamoeba, or the more common way is through the use of contact lenses. Ayan, so sa mga tig-contacts dyan, please be careful with my heart. Charot. Again, please be careful with using your contact lenses. Okay. Now, how do contact lenses, or asa siya nakuha? It's again contaminated saline, no? Homemade saline na contaminated with your amoeba, and then dito nimo gibutay imong contact lens, or again, poor imong hygiene sa contact lens, or nanay daay mga abrasions or ulcerations imong cornea. So again, pwedeng trauma siya, but ang main method good is through the use of contact lenses. So the contact lens acts as a mechanical vector. It acts as a mechanical vector, okay, because again, it assists in the transport of the amoeba from the container of your contact lens to the eyes. Okay, so ayan, it can lead to keratitis. So your, again, cornea serves as a mechanical vector. So your mechanical vectors can be living things or non-living things. Okay, alright. Basta ang purpose niya is mo transport sa organism or sa parasite from one host to another, or from one area to another. Okay, alright, ayan. Now, Acanthamoeba keratitis can also be secondary or opportunistic lang to your herpes simplex or viral na keratitis. So because ang first na disease kay herpes simplex, so pwedeng ang matambalan ra is the virus, no, the viral infection. Ang Acanthamoeba dili matambalan, so pwede siyang mo padayon gyapon ng infection. Okay, so secondary siya. It could be secondary or opportunistic to patients with herpes simplex keratitis. Okay, and again, as mentioned, it's not through contaminated water, okay, but contaminated saline. Okay, so kana mga homemade saline solutions for those that are using contact lenses. Okay, all right, so very important that if you use contact lenses, dears, make sure that you follow your ophthalmologist's prescription, and if na siya gihatag yun na solution sa imong contact lens, wag ito gamito nimo. Okay, and then wash your hands while before applying and after applying pod para at least less ang contamination. Okay, all right, ayan. Now, for Acanthamoeba also, Acanthamoeba, di ba, they have similar characteristics with the gram-neg bacteria Pseudomonas, di ba? Because Pseudomonas is also well known to cause keratitis. Okay, but they are not recovered at the same time from the same patient because it is believed that Pseudomonas inhibits the activity of Acanthamoeba. Pwedeng iyahang gina-inhibit ng activity sa Acanthamoeba, pwedeng dili maglihok Acanthamoeba. Could be because of competition, no, sa area, so mas paspas mo grow ang Pseudomonas kaysa sa Acanthamoeba, so overthrow niya ang imuhang Acanthamoeba. So makadaog siya against Acanthamoeba, ganun. Okay. Now for symptoms, severe ocular pain and vision problems. And if you examine the cornea or infected cornea, you may see again trophozoites and cysts. Now if you do not address this, it may lead to corneal perforation, mabusot imong cornea, and may lead to loss of vision. So it's important again that you, once you present with these symptoms, okay, and then ay mga appearance pag yun sa imong eyes, magpa-check jud ka dayon, okay, para maagapan agad-agad. Okay, now remember again, very important note, Acanthamoeba species are well accepted opportunistic pathogens in patients with AIDS or immunocompromised patients. Okay, so usually they present as cutaneous lesions, no, mga sakit sa skin, all right? But again, they are opportunistic pathogens among these patients. Okay, all right, so that's for the diseases that your Acanthamoeba may cause. Okay. So first is, of course, again, your Acanthamoeba keratitis. So as you can see, di ba, parang na-ring-like na na-form, ring, di ba, na murag infection. So murag iyang gitrace ang contact lens kung asa nimo gibutang, di ba? So once inani na ang makita sa imong ophtha, okay, or sa doctor, then possible na pwede na siyang idea na, okay, that is Acanthamoeba keratitis. All right? And this is your cutaneous lesion from a patient, immunocompromised patient that is affected by your Acanthamoeba. So cutaneous lesion. Okay? All right. Ayan. So usually seen again in immunocompromised patients or those with AIDS. Okay. All right. So that's for the diseases that your Acanthamoeba causes. Okay? All right. Ayan. Sige. Now we go now to your diagnosis. How do we diagnose this free-living amoeba? Again, similar lang with your Naegleria fowleri, no? If sa CSF, centrifugation, smear gapon, and then look into the possible presence of trophozoites and cysts, diba? Because again, for Acanthamoeba, pwedeng trophozoites and cysts imong makita. All right. Now for culture, again, non-nutrient agar pa din, which contains E. coli, but this time you use page saline. So Acanthamoeba, dears, no, it can tolerate, okay, it can tolerate sodium concentration or NaCl concentration of 0.85%. So unlike sa imong Naegleria fowleri na dili mo tubo if greater than 0.4% ng NaCl, your Acanthamoeba can grow even if the NaCl concentration is 0.85%. So please take note. All right, ayan. Next, for tissue stains, if you want to stain, no, from histological preparations, you may use periodic acid shift. It can stain the cyst wall red, and methenamine silver, which stains the cyst wall black. So you could also, another stain the ideas na wala na ko nabutang is calcofluor white. Okay, calcofluor white. All right, ayan. But in ocular samples, usually identification may be difficult because it can mimic or it can look like your keratoplasts or cells of the cornea, or even neutrophils and monocytes. So it needs experience, it needs training to identify. Okay, so if the doctor or maglisod ang ang examiner sa pag-examine or pag-identify, then pwede siyang look into history, no, of the patient and presentation of symptoms pa rin. Okay? All right. Ayan. And of course, CSF, if naigae or BAL, bronchoalveolar lavage, no, for respiratory symptoms. So kanina mga specimens atong gamiton. All right. But for keratitis, again, our specimen of choice sa man to is corneal scrapings. Please take note. So mukuha ta sa surface of the cornea, atong scrape gamay, okay, and then examine. So specimen of choice again for keratitis, Entamoeba keratitis is corneal scrapings. All right. Please take note anang BAL dears ha. Nigo sa boards, previous boards not on our time. Sa histopath na subject, ang question kay what is a BAL specimen? What type of specimen is BAL? BAL lang. So is it respiratory? Is it blood ba to? Is it so pa to other choices? So of course it's a respiratory specimen. So dala pa lang daan 1 point na ka, o di ba? So meaning of BAL, bronchoalveolar lavage. Okay, ayan, sige. So that's for Entamoeba species. So here is some pictures, no? Entamoeba species na cyst, gistain og pas, di ba? It's color red, okay? Color red. Actually, na siya red, pero marag reddish purple, okay? And for methenamine silver, black. Ayan, black ang cyst wall, okay? And as you can see again, ang iyahang inner cyst wall, dears, dili gud siya circle na perfect, no? It has a lot of shapes, polyhedral, okay? Ayan. And of course, again, ang color for PAS, red or reddish purple, and for methenamine silver, black. Okay, all right. Ayan. So before we continue to the next, hapit na mahuman, your Balamuthia, do you have any questions, dears? Okay. Right. Okay. All right. Ayan. Sige. Again, no? So ito yung characteristic na cyst na itong emphasize good for Entamoeba. Double wall, okay? Outer wrinkled cyst wall. Okay, all right, ayan. So very important to take note. Do not forget, okay? Ayan, sige. All right. Sige, any questions before we continue? Okay. All right, sige. So we'll now continue with the next, no? The next free-living amoeba is Balamuthia mandularis. Now, Balamuthia mandularis, similar again with Entamoeba na life cycle, and in the fact that they don't have a flagellated stage. So, di ba? Same rin siya with Entamoeba species. So the difference lang is there could be a lot of nucleoli sa nucleus ay muhang Balamuthia. But again, seldom rin siya makita or medyo difficult to see under preparations. Okay, so kailangan siya o further tests pag yun to really specify. Okay, ayan. Now, for both trophozoites, again, and cyst can be recovered, okay, in the tissues. Parehas sa imuhang Entamoeba. Okay, now we go now to your trophozoites. Again, trophozoites irregular in shape, okay, and very important characteristic, iyang pseudopods, okay. Pseudopodia, broad pseudopods, finger-like iyahang pseudopodia. So dira pa lang daan difference na sa trophozoites with your Entamoeba. Because your Entamoeba, di ba, spine-like pseudopods, Acanthopodia. For Balamuthia, finger-like or broad pseudopodia. So mga mahilig mag-fingers dyan, charot. Okay, mga finger-like pseudopodia. Okay. Now for the cysts, as you can see, same siya na na-eye wrinkled, no? Outer cyst wall, parehas Entamoeba. Pero this time, 3 layers iyahang cyst wall using electron microscopy. So you have the outer wrinkled ectocyst, you have the mesocyst na middle layer, and then you have the inner endocyst na medyo dili makita. So if you use light microscopy lang, your bright field microscopy, duhara ka layers yung makita or 2 walls. Alright, so if ang question sa inyong exam, kung asa man gani na exam, no, sa med ba, sa boards or whatever, ang question kay 3 layers of cyst wall and the outer is wrinkled, then ang atong answer is Balamuthia. But if ang question kay double wall, and the outer wall is wrinkled, then Entamoeba atong answer. Pero if ang question kay wala gi-specify kung double ba or 3 layers ang wall, ang giingon ra kay outer wall is wrinkled, then ang atong answer ana is Entamoeba. Okay? Because Entamoeba ang more common. Okay? Ayan. So, yan atong mga mga techniques na to, mga tactics ha. So, ayan. So, basta look at the question lang, how it is phrased. Okay? Alright, ayan. Sige. So, for Balamuthia manggod, it's medyo recently lang siya na establish na makakosog infection. Okay? So, but similar siya with the Entamoeba. So, ayan. That's why we need to, in a way, differentiate. Alright? Okay, ayan. So, here are some pictures. So, this is the trophozoite of your Balamuthia. So, di ba, broad, no? Meron siyang tree branch, no? But it's finger-like, meron siyang fingers, and broad imuhang pseudopodia. And you have the cyst, again, you have the outer wrinkled, then the mesocyst, and the inner endocyst. Again, 3 layers siya if electron microscopy mong gamiton. Pero kung light microscopy or normal microscopes, duha ra siya ka-wall. Okay, all right. So that's for Balamuthia mandularis. Now for the disease, very similar with your GAE. Again, not associated with swimming in fresh water. Now for diagnosis, for culture, dili siya mo differ, dili siya mo grow in parehas with Acanthamoeba or Naegleria using non-nutrient agar with E. coli. Okay, because ang iyang ganaan is live cells, no? Mammalian cell culture parehas sa iyaha, parehas sa mga viruses, no? Inana siya ka-choosy, no? Inana siya ka-choosy burnout. Charot. Again, this is mammalian cell cultures. Okay, live cells iyang kailangan ganahan for culture parehas with viruses. Okay, all right, ayan. Now for definitive identification, we use electron microscopy and histochemical methods, usually through immunofluorescence na test. So for definitive diagnosis of this organism, DIRS, no, again, it's not routinely performed. Electron microscopy and immunohistochemical tests, no, immunofluorescence. So again, for speciation, for accurate speciation, usually reference labs, research labs, high-end hospitals if they offer, silang pwede maka-perform ani. But if routine lang, no, kanang sa ato ragong routine hospitals, it's less likely yun na ma-perform. Okay, we don't have the capability of that for that. Okay, all right. So that's for the diagnosis of Balamuthia mandularis. So here are some pictures, no, in tissue. So as you can see, outer wrinkled cyst pa rin siya. Pero ang inner, no, ang inner wall niya is spherical this time, no? So one of the differences put with Acanthamoeba, because ang cyst sa Acanthamoeba, di ba? If you can remember, wrinkled yung outer wall, pero iyahang inner kay polyhedral, meaning mo assume siya a lot of shape, di ba? Ayan. So that's for Balamuthia mandularis in tissue. And this is Balamuthia mandularis in indirect immunofluorescence assay, di ba? Kani mga ni Fleuris ug apple green mo ni siya ang Balamuthia na organisms. Okay? So this is from a brain tissue, I believe, and kani mga ni Fleuris mo ni siya ang Balamuthia na organisms. Okay? So again, the methods for definitive diagnosis again are not performed routinely. So mas best na ang maka-perform is mga research labs or reference labs. Okay. All right, ayan. So that's for Balamuthia mandularis. Now for the last free-living amoebae, no, these are just mga recently discovered na mga free-living amoebae that can cause infection in humans, pero usually mga isolated cases lang. All right. So first is si Sapinia spp. So you have Sapinia pedata. It was recovered from a patient, 38 years old, no, nag exhibit og mga meningitis na symptoms, frontal headache, blurry vision after a sinus infection. And pag lantaw sa CSF or pag excise sa iyang brain lesion, nakakita silag sa Pinia pedata, a free-living amoeba that is found in soil contaminated with feces of elk and buffalo. But again, isolated cases lang, dili kayo common. Mga recently lang na-discover sa Pinia pedata. The next is sa Pinia diploidea. So sa Pinia diploidea, trophozoites, characteristic niya contains 2 nuclei and it's combined with a perpendicular filament. So na siya filament na nag-combine or nag-connect sa duha ka 2 nuclei. Okay? And it can ingest blood cells and stain brightly with GEMSA or periodic acid shift na mga stains. Sa Pinia diploidea. And the last one is Paravalcamphilia francini. So again, isolated case. It was found in a CSF of a patient that had symptoms typical of PAM. So abi nilang Naegleria fowleri, but paglantaw nila, it's another type of free-living amoeba, your Paravalcamphilia francini. But again, these free-living amoeba, other free-living amoebae, these are again isolated cases. Recently lang sila na-establish that can cause infection. And rare po to happen, not common na ma-isolate or ma-recover. Okay, but once they infect humans or once they are transmitted to humans, they can cause infections. Okay, all right. So here is an example of Sapinia diploidea. So here are the 2 nuclei, and then the center is the filament, okay, that connects the perpendicular filament that connects the 2 nuclei. Okay, and for stained in tissue, so two nuclei and then a filament that connects the two nuclei together. Okay, so that's for the other free living amoebae. And all for that's all for free living amoebae. Okay, so before we proceed to the last topic for today, your subphylum Ciliophora or Ciliata. Yes, do you have any questions, dears? Okay. So bye-bye na to ng mga free living amoebae, no? So again, kinsa tong amoebae, no? Free living amoebae that contains two nuclei attached by a perpendicular filament. Kinsa to siya? See? Two nuclei attached by a perpendicular filament. You have
Sapinia diploidea.
okay. All right. Very good. Sapinia diploidea. All right. And again, three layers ang wall seen in electron microscopy. The outer is wrinkled, and then you have a mesocyst and an inner endocyst.
Balamus.
Balamuthia.
All right. All right. Balamuthia mandularis. Okay, don't forget. Pero kung double wall ra, wrinkled outer cyst,
Entamoeba.
okay.
Entamoeba.
Wag naman magalit. Okay, so Entamoeba. All
Sorry.
right. Kalma lang. Okay ra. All right, so Entamoeba. Okay, all right. Now for the last no, last na subphylum is of course your class subphylum Ciliophora ciliata. So these organisms of course contains cilia, okay, which is their locomotory organelles. So this is what they use para makalihok sila. All right, so these are short extensions of the cytoplasm that cover the surface of the organism. These organisms also contain or possess two nuclei, a large macronucleus and one or more micronuclei na mga gagmay. All right, now in the environment, no, protozoa, daghang ciliates. Okay, there are a lot of ciliates. A very good example, di ba, si Paramecium. You also have mga stentor, mga other ciliates pag yun. And these ciliates are, again, non-pathogenic and non-parasitic because there's only one, and that is your Balantidium coli. Nagbubukod tangi, siya lang talaga. Sana all. Okay? So siya lang mag-isa. Okay? The only human pathogen among the ciliates. Okay? And infection is also rare sa mga US, no? Sa mga first world countries. But in the Philippines, this is quite common, no? Pwede siyang ma-recover yung mga Balantidium coli. Okay. Now, aside from being the only pathogenic ciliate, it's also the largest, ayan, ako mga characteristic talaga, mga press the buzzer, characteristics ni Balantidium coli. Largest protozoan parasite affecting man. So, ayan, kanang mga question, no? The only pathogenic ciliate, the largest protozoan affecting man, ako bang na mag-isip, it's no other than Balantidium coli. Okay, so Balantidium coli. All right. Now, for the mode of transmission, same with your amoeba, ingestion of fecally contaminated food or water that contains your cysts. Okay, and the food or water contaminated siya with the feces of pigs, hogs, HOGS, or swine, basta sa pigs. Okay, because the natural host of this organism or Balantidium is of course your pigs. So humans are accidental hosts lang. So again, acquired through accidental ingestion. Again, ingestion of food, water that may contain the cysts. Okay, reservoirs could be your pigs, monkeys, and even rats. And the infective stage pa rin is still the cysts. Okay, so that's for Balantidium coli. Okay, now we go now to your morphology, starting first with the cyst, trophozoite, sorry. So for the trophozoites, again, di ba, largest protozoan, so large po diyahang trophozoite. So as you can see, 50 to 100 micrometers in length and 40 to 70 micrometers in width. And it resembles daw a sack, no, marsyag sako or sack in shape. So wala na ako siya nabutang dagay dira there, so please write na lang. And Balantidium itself, the name, it means little bag, di ba? So ka-cute. So Balantidium, little bag. Okay, ayan. So sack in shape. All right. Now, very important, the anterior end is quite pointed and the end, the posterior or sa likod, kay medyo gifted na siya, no? Broadly rounded na siya. Okay, now the anterior end contains a cytostome, which is the mouth. So, dira siya mo kaon or dira mo sulod iyang food. And the posterior end contains the cytopage or cytopage, which is your anus. So, diri mo gawas ang mga wastes niya. Okay, so this is the illustration of your trophozoite. Again, cilia in the surface, and you have the cytostome and the cytopage or cytopage. So this is a stained image, no? Again, you have the cilia, if makita, no? And the cytostome nagi-point sa arrow, which is again the mouth of your Balantidium coli na trophozoite. Okay, now continuation again, the trophozoite cytoplasm, granular, vacuolated that may contain ingested bacteria and debris. And very important, distinct characteristic are the nuclei. No, a large macronucleus that is described to have a kidney or bean-shaped, no, bean-shaped, kidney-shaped macronucleus, and a dot-like micronucleus. So your macronucleus functions as a vegetative for vegetative functions like metabolism, no, daily processes sa imuhang organism, whereas your micronucleus serves as a sexual organ or for sexual reproduction. Okay? Motility is described as thrown ball, so magilabay na bula, or rotary, rotates, and boring. So meaning parang nagbuslot siya, no? It's like making a hole. Ah, yes, so boring. Okay, ayan. So here's an example, no? Using your wet mount, again, very characteristic, very distinct characteristic, the kidney-shaped, bean-shaped macronucleus of your Balantidium coli. And very important, your cilia, na klaro kaayo. And again, another stained preparation, again, the cytostome, the oral apparatus, no, the mouth, and very important macronucleus, kidney-shaped or bean-shaped macronucleus. Okay, cytoplasm is quite granular, vacuolated, again, kani vacuolated, that may contain, again, ingested bacteria or debris. Okay, that's for the trophozoites of your Balantidium coli. Now, for the cyst, ayan, so spherical, oval, no, may contain mga vacuoles pa rin. It could have a double wall, even, especially in young cysts, may contain a row of cilia na nagdevelop pa lang. And stained cyst, again, ang very important characteristic ang makita ra is the macronucleus. The micronucleus usually dili gyud siya makita. All right? That's why we look into the macronucleus because muna siya maklaro. Okay? All right. The large macronucleus, again described as having a kidney shape or bean shape. Ayan. All right. Okay. So here's an example. Again, using your iodine stain, di ba? Very claro, no? Your kidney shape, bean shape macronucleus, and kani mga circles here are the vacuoles. All right. And again, stained pa rin, preparation, very claro good is the large kidney shaped, bean shaped macronucleus. So kana palang daan, klaro na, perfect na, no? Press the buzzer kay Balantidium coli gid siya. Okay, all right. Yeah. So that's for the B. coli na trophozoite and cysts. Now we go now to the disease, no? Of course, the disease is termed balantidiasis, okay? Diba? Mga iasis, okay? Pronounce it correctly, okay? Balantidiasis, jardiasis, no, okay, ayan. So, balantidiasis, it resembles amoebic dysentery. So, pwede siyang mo-appear as amoebic dysentery. So, magkalibanga, and the stool contains pus, mucus, and blood. And aside from that, the organism can also cause ulcers, okay, in the mucosa and submucosa of the large intestine. But as you can see, mas daghang stools ang pwedeng mapagawa sa patient in a day, 15, no? For amoeba or for amoebiasis, usually mga 10 lang. So imagine 15, makaluya na siya kaayo, okay? That's why because of 15 liquid stools, tremendous fluid loss, it may be similar to a diarrhea caused by cholera or your Vibrio and some coccidial or microsporidial infections. Okay, so very important that you replenish those mga fluid loss nimo to prevent dehydration and electrolyte imbalance. Okay, all right, ayan. Now, again, as mentioned, it has the potential to invade tissue because it has this enzyme, hyaluronidase. Okay, please take note of that, dears. All right, and it may cause, again, ulcers. All right. It may vary in shape, but the general shape niya is rounded base and wide ang neck. So it may appear like a beaker. Okay, so compared sa imong Entamoeba histolytica na flask-shaped, ang imuhang B. coli kay beaker-like. So wide ang neck, wide pod ang bottom or rounded base pod siya. Okay? Whereas ang E. histolytica, narrow ang neck, rounded base, diba? So mura siya Erlin Mayer flask. But for B. coli, again, wide ang neck, rounded ang base. So mura siya beaker. Medyo dako siya or tambok iyang appearance. Okay? All right. It may spread to other tissue or it may invade other tissues, but it's not common, dili kay common ang extraintestinal diseases for balantidiasis. Okay? All right, ayan. And this, dears, wala na ako nabutang dira, the presence of salmonella, specifically salmonella gyud, it can aggravate your balantidiasis because it can infect the ulcers. Okay? So, actually, other gram-neck bacilli pwede pod, but for Belisario, yan yung specify na salmonella. Okay? So, salmonella na bacteria, once the patient also has salmonella in the intestines and then nasa Balantidium coli na ulcers, pwedeng mo grabe iyahang Balantidium coli infection or balantidiasis because salmonella may infect the ulcers made by Balantidium coli. Okay? All right. And here's an example of a biopsy, no, from the intestine that contains B. coli. So as you can see, diba, when stained, ang klaro gyud is the large macronucleus na bean shaped. Okay, so very, very klaro gyud. Okay, the large kidney shaped or bean shaped macronucleus. Okay, all right, that's for balantidiasis. All right. Now for diagnosis, very important is still again routine stool exam. Now it's recommended that you examine it in a wet preparation from a fresh or concentrated material. Okay, so wet prep. So direct fecal smear gyud ang imuhang ang recommended. Okay, because in permanent stain smears pwede siyang ma-stain very darkly and may obscure or dili makita ng internal morphology. So dili makita tong large macronucleus, diba? So it will make your identification difficult. So that's why it's recommended na fresh wet prep, no, from direct fecal smear so that you can really see the trophozoite, no, and the cyst, and especially the inclusions. All right, because in permanent stain smears using our stool stains like trichrome or iron hematoxylin, it may stain very darkly. Okay, so dili maklaro ang internal morphology, so which leads to difficulty in identification of the organism. All right, okay, and aside from that, also it can be confused with helminth eggs, especially if walay cilia, because it could mimic, no, a helminth egg, especially with its large size. All right, so samot na if walay cilia pero na table. Charot. Ay, may ganun. Pangit talaga ng joke. Gipush Japon ako siya. Okay, so basta walay cilia, okay? Walay cilia, it can be mistaken as a helminth egg. Okay, so muna siya mga challenges in identifying Balantidium coli. So here's an example, no? This is stained using trichrome, diba? As you can see, it takes up much of the stain and stained very darkly, no? Although maklaro ang cilia ani, but your large macronucleus, which will really confirm our diagnosis, diba? Or our identification, it cannot be seen. So medyo lisod gyud siya. All right. Now, this is an example of a cyst of Balantidium coli na walay cilia. So it could again mimic, or it could look like a nematode egg or a helminth egg, mga trematode egg, diba? Okay, wala siya cilia pero na ay table. So i-push gyud ako ni na joke. My gosh. Okay, all right. So please katawa mo dear, reply mo ha 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 sa chat. Charot. Okay, joke lang. Okay, all right. So ayan, so these are mga how we identify your B. coli. What we need to take note lang yun in identification is the presence of a large kidney-shaped macronucleus. Okay, mudyo na siya. Okay, both in trophozoites and the cyst. Okay, all right, and that's the end. Yes, that's the end of the first group of protozoa na lecture, both your amoebae and your ciliate. Okay, all right. So before we end, dears, yes, do you have any questions before we end? I'll say stop ang recording if okay ra ninyo. Okay.